Hello, I'm Alan Utter, the Provost here at Arkansas State University, and I would like to officially welcome you to our annual Create at State event, in which we have the opportunity to celebrate faculty-mentored student research and creative endeavors. Our goal here at Arkansas State University is to provide every student who wants to be involved in a research project the opportunity to do so. Over the next 3 days, 241 of our students will deliver 190 original research presentations that have been mentored by 60 of our individual faculty members. I want to first wish each and every one of our students the very best of luck with your presentations over the next few days. I also want to take a moment and thank each of our individual 60 faculty mentors that have taken time out of your schedule to work with our students on these original research and creative endeavors projects. I also want to thank our faculty that have served as reviewers of the abstracts that are going to be presented at the meeting. And also I want to thank the 85 individuals that have agreed to serve as judges for the award competitions at Create at State. In closing, I wish everyone a great meeting. I look forward to listening to the new knowledge, to the new discoveries that have been made on our campus this past year. Have a great meeting, and wolves up.
Hello, I am Lynn Boyd. I am the Dean of the College of Sciences and Mathematics, and I'm here to welcome you to Create at State. Create at State is something we do every year at Arkansas State University to celebrate the research and creativity that's happening here at A-State. In the College of Sciences and Mathematics, we love doing research and trying to understand how the natural world works. So we're continually making new discoveries and adding to the body of knowledge. I like to tell students that all of those things that they read about in their textbooks and those pictures and diagrams are the result of curious people who wanted to ask questions about how the world works. And they did research and made discoveries, and now that constitutes our body of knowledge and what we can convey to students when they take our classes. But we have faculty and students here at Arkansas State University that are doing really interesting research, really exciting stuff. I'm really excited to hear from our students the creative research that they've been doing, the interesting questions that they've been asking, and then the discoveries that they've made. So with that, let's move on to the student presentations. Thank you.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello everybody, my name is Izzuddin Ahmed. I'm a junior biologist student at Arkansas State University, and today I'll be presenting my project, which is the effect of root biomass on peanut stibonite yield in hairy cultures of the peanut. So a little bit about what oxidative stress is before I dive into my project. Oxidative stress is caused by free radicals that are in our bodies. These free radicals cause us a host of different health issues, such as heart attacks, strokes, and Alzheimer's, as well as the diseases that you see on the screen. So what are stilbenos? Stilbenos are polyphenolic compounds that are produced by various plants, such as grapes and peanut. These stilbenos have a host of different health benefits, such as neuroprotective abilities, anti-cancerous, and antioxidant activities. If you ever heard about the quote, a cup of wine away keeps the doctor away, it's because wine has a special kind of stilbenos called resveratrol. And resveratrol has been heavily commercialized over the past few years. The problem with resveratrol, however, is that it produces this, it has a low bioavailability when it enters our body. Fortunately for us, our lab has been able to produce this phenylated stilbenoids such as arachidine 1, arachidine 2, arachidine 3, and arachidine 5, which has a phenyl tail to them, which potentially allows them to stay in the body for a longer period of time. And we produce these stilbenoids from our system, which is the hairy root system. And to that end, my project is to increase the hairy root biomass, and I believe that will lead to an increase in stilbenoid yield upon extraction at the mid-log phase. To test that hypothesis, I established a growth curve at the 500 mil growth curve in the 500 mil flasks. I elicited the hairy roots at the mid-log phase. I extracted my stilbenoids from the culture medium, and I analyzed the stilbenoids by using HPLC, which is high performance liquid chromatography. So my methods for the growth curve and the elicitation and extraction are listed in the screen. So what I did for the growth curve is every 3 days, I took 3 flasks, froze them in the -80 freezer, lyophilized them for 3 days, and then I analyzed the dry weight to establish the curve. For my elicitation and extraction, I took 3 flasks at the day 12, which you will see why later on. And I elicited them using the elicitors hydrogen peroxide, methyl jasmonate, magnesium chloride, and cyclodextrin. And I did a standard organic extraction using ethyl acetate, and then I run the extract in HPLC. So my results, which here is why I elicited them in day 12, because day 12 from the growth curve is the mid-log phase, which is where we produce the most stilbenoids out of any other days in the curve. And to that effect, we can see the HPLC results down here, which is the extract composition, where we see an exponential increase in stilbenoids when we grow them in the larger flask, as well as the HPLC profile show you that this peak right here shows a jump in larger flasks, as well as that's arachidine 3, as well as arachidine 1 right here. And that's the results for the Hall 3A phase, the Hall 3A line. For the Georgia Green line, we see a much more comprehensive increase. In terms of in the growth curve, we did delay the growth curve a little bit because the Georgia Green line grows a little bit slower. So the mid-log phase is day 15. And my extract composition shows you how much more stilbenoids we're getting in the larger flask, which is way more than we expected. And the HPLC profile show you just how much more stilbenoids we're getting in the larger flask. So to that effect, my conclusion is an increase in root biomass correlates to an increase in stilbenoid yield in the extract, and culturing procedure, which is the 500 ml flask with 100 ml media, results will be tested with other peanut cultivars, such as the TIF runner, for future work. To that end, I would like to thank Dr. Fabricio Medina Bolivar for giving me the opportunity to work in his lab, the Arkansas Biosciences Institute for giving me the opportunity to work in a research lab, Arkansas State University, and Create A-State for giving me this platform. And finally, last but not least, I would like to thank you for listening to my presentation today. Have a great day, and Red Wolves.
Welcome to Create at A-State. My name is Jonna Berryhill, and I'm going to spend the next 5 minutes talking to you about identifying major depression patient subgroups based on neuropsychiatric symptoms. Major depressive disorder, or MDD, is a common, often disabling condition affecting over 300 million individuals worldwide. Clinical symptoms are insufficient to guide appropriate treatment selection, and presently, treatments are therefore selected empirically, relying on a trial and error approach. In this study, we have profiled by mass spectrometry the serum metabolites of 290 unipolar patients from Mayo NIH PGRN AMP study with major depressive disorder. Blood samples were collected at baseline after 4 and 8 weeks of antidepressant treatment. The 17-item Hamilton Depression Rating Scale and the 16-item Quick Inventory of Depressive Symptomology Scale were used to gauge their depression symptoms and overall depression severity. Using multivariate and univariate statistics, we 1. identified patient clusters based on their symptom similarities. 2, identified the patients within each cluster who responded to or failed the treatment. And 3, characterized each cluster based on symptoms of anxiety and insomnia. The next couple slides, you'll see our results. We observed 3 clusters at each of the time points and represented the clusters in terms of treatment response. The following plots show the overall depressive symptom trajectories in patients who responded to versus failed treatment. If you look to the plot on your left, there you will see the responders. These are the patients who showed at least a 50% reduction in their total depression symptom scores from baseline to exit. The plot on the right shows the complete non-responders, or the treatment failures. These patients had less than 30% reduction in their depression symptom severity from baseline to exit. Notice how the non-responders tend to start with the higher score, meaning more severe depression symptoms, and end with the higher score. Next, we characterize patient clusters in terms of the symptoms of anxiety. If you look at the colored lines on these plots, you may see a triangle pattern or odd lines. This is because the score variables are discrete. You should focus on the trend line instead. Once again, the responders are on the left and the non-responders are on the right. Notice how anxiety symptoms in the responders showed a significant decrease after just 4 weeks of treatment, while the non-responders actually had increased anxiety while being treated for depression. Next, we characterize patient clusters in terms of the symptoms of insomnia, or lack of sleep. Once again, focus on the trend line when reading these plots. Responders tend to have a significant decrease in insomnia symptoms halfway through treatment, while non-responders showed little to no decrease in insomnia throughout treatment. Our results indicate that identifying and characterizing patient subgroups will help in selecting proper treatment, predicting treatment response, and understanding disease mechanism. After analyzing the data, patients who come in with less severe depression symptoms at baseline are more likely to be in remission after 8 weeks of treatment. As early as week 4, patients whose anxiety symptoms do not decrease are likely to not respond to treatment. We also notice a similar trend in patient insomnia symptoms. If insomnia symptoms have not decreased by week 4, patients are likely to be treatment failures. In the future, we plan to 1. Characterize the patient clusters in terms of all other depression symptoms, and 2. Correlate the depression symptoms to the patient blood markers in each patient cluster. This will help physicians diagnose major depression patients more objectively through a clinical test without using a trial and error method. Thank you.
Hello everyone, my name is Lin Chi Ho and I'm a senior biology major here at Arkansas State University. I have the privilege to work at the Arkansas Biosciences Institute in the Medina Boulevard lab with another student named Zebedee Mohammed Hosenpour. The title of my presentation today is the anti-cancer effect of prenulated stilbenoids from peanut in combination with paclitaxel in triple-negative breast cancer cells. Breast cancer is the most common cancer in women worldwide, and triple-negative breast cancer, or TNBC, consists of 15% of breast cancers. It's one of the deadliest types because it's lacking in 3 receptors that most hormonal treatments target in order to stop the spread of cancer. Because of this, TNBC has few treatment options available, so there's an ongoing search for new treatments. A common treatment used to treat TNBC and other cancers is chemotherapy with the use of a drug called paclitaxel. However, during the use of paclitaxel, multiple undesirable side effects are observed. To reduce these side effects, natural products called phenolic compounds could be used in conjunction with paclitaxel. When plants such as peanut are stressed, they produce these defensive compounds, which have been found to possess properties like anti-cancer. Prenulated stilbenoids such as arachidin have this extra perennial tail attached to their primary structure, and this extra piece results in a potential higher anti-cancer activity. Thus, the goal of this research is to evaluate the anti-cancer property of these prenulated stilbenoids as natural products for the treatment of TNBC when in combination with paclitaxel. My hypothesis is that the treatment of TNBC cells with paclitaxel in combination with the prenulated stilbenoids arachidin 1 and arachidin 3 will be more effective in inhibiting cell growth or mitosis compared to treatments of paclitaxel and prenylate stabenoids alone or with resveratrol. In this research, my objectives are to compare cell cycle events of TNBC cells after treatment of arachnid-1, arachnid-3, and resveratrol using flow cytometry, and to evaluate the IC50, which is the concentration of compounds that will inhibit the growth of 50% of cells of paclitaxel and the 3 stabenoids alone and in combination with each other on TNBC cells using cell viability assays. To induce the production of stabenoids, peanut hairy roots, an immortalized bioproduction system, were treated with 4 different elicitors: cyclodextrin, methyljasminate, magnesium chloride, and hydrogen peroxide. Once the stabenoids were produced in the culture media, they were separated via semi-perforative high-performance liquid chromatography, or HPLC, so that the stabenoids were purified for testing. Using flow cytometry, the number of cells from the TNBC cell line MDA and B231 in each stage of the cell cycle can be observed. In the G0 and G1 phases of the cell cycle, cells are undergoing normal cellular functions. In the S phase, cells begin to prepare for division, and so they replicate their DNA. In the G2 phase, cells continue to grow and prepare for mitosis or nuclear division, which results in cell division. Since I'm dealing with cancer cells, they divide uncontrollably, so I want to prevent cells from undergoing mitosis. 3 days after treating the cells with varying concentrations of the 3 stabenoids, it can be seen that at 50 micromolar concentrations, arachnid-1 and arachnid-3 have more cells in the G0 and G1 phases compared to those cells treated with resveratrol. Because of this, we want to focus on treatments of lower concentrations since they're more effective. Cell viability assays can be used to show how many cells are still alive and growing after being subjected to compounds. They help to determine desired concentrations of the compounds being tested. The results from the cell viability assays show that after 3 days of treating the TNBC cells, the IC50 of paclitaxel decreased when in combination with resveratrol, arachnid-1, and arachnid-3. However, less arachnid-1 and arachnid-3 were needed to achieve this result compared to resveratrol. This shows that prenylate stabenoids are more effective in killing or inhibiting cancer cells compared to the non-prenylate stabenoids, and they can be used to lower the concentration of paclitaxel needed, which is good due to its toxicity. From the results, it can be concluded that the prenylate stabenoids arachnid-1 and arachnid-3 at lower concentrations are more effective in preventing TNBC cells from undergoing mitosis compared to the non-prenylate stabenoid resveratrol. This means that prenylate stabenoids potentially have higher anti-cancer properties than non-prenylate stabenoids. The IC50 of paclitaxel and of the stabenoids also show a decrease when in combination with each other, particularly when paclitaxel is combined with prenylate stabenoids. This shows that when mixed with arachidins, paclitaxel can be used in lower concentrations to inhibit the same number of cancer cells. If the doses of paclitaxel could be lowered, this may then result in diminishing some of its undesired side effects. Before I end my presentation, I'd like to thank you all for the opportunity to present at Create Estate and for giving me your time and attention today.
My name is Michaela Stone, and I'm presenting the assessment of an herbal compound as a potential novel treatment for migraine. The purpose of this study was to look at the effect and mechanisms of action of a proprietary mixed Chinese herbal extract that was effectively used in humans, and we did that using a rodent migraine model. This is important because migraine is a very common disorder and it affects females twice as much as it does males. Our migraine model consists of injecting female CD1 mice with inflammatory mediator, or IM, onto the dural surface. We inject 5 microliters at a 2x concentration, and this is to imitate the effects of the migraine in humans. To determine whether or not tactile allodynia developed, we use von Frey filaments on the periorbital and hindpaw regions. If the mice respond positively, it indicates that they are feeling pain in that area. A positive response would look like the retraction of the hind paw, slapping of their face, or attacking the filament, among other things. With these filaments, the mice are tested prior to injection so that we have a baseline response frequency, and then also every hour for 5 hours after the IM injection. Here, picture A shows the von Frey filaments, and the filaments themselves are made of nylon. Picture B shows how we test the animals. They are placed in chambers on top of wire mesh so that we can access both the periorbital and hind paw regions without otherwise disturbing them. And then picture C shows how we obtain the standardized testing force. So at the point that the filament bends, a certain force is being exerted. We use 0.4 grams on the periorbital region and 1 gram on the hind paw. And then we record response frequencies out of 10 applications. Both of these figures show the mouse response frequencies over 5 hours after injections. These figures show that our IM solution does produce the tactile allodynia in both the periorbital and hind paw regions, and that is represented by the blue lines on both graphs. This is compared to injecting synthetic interstitial fluid, or SIF, which does not produce the tactile allodynia and is represented by the orange lines on both graphs. These graphs compare the injection of the IM alone to the injection of the IM and administration of the herbal compound. The compound was given orally at a concentration of 80 milligrams per kilogram twice a day for 2 days, and then again half an hour before giving the IM injection on the 3rd day. The blue line represents the IM only, and the orange represents the IM along with the drug administration. So as you can see, the mice that received the herbal compound still developed a significant amount of tactile allodynia in both of the tested regions. So we unfortunately discovered that the dose of herbal compound that we were using did not block the migraine-related pain induced by the IM. So our next steps will be to adopt a different migraine model, possibly by giving rats inflammatory reagent and then provoking them with human migraine triggers. We may also change the dosing regimen of the compound by changing the individual doses, the number of doses, or by changing the way we administer the compound. Lastly, I would like to acknowledge Dr. Jennifer Shih for allowing me the opportunity to do research in her lab and providing me with the tools to do so. Krishna Sharma and Mehul Shah for teaching me the injections and behavior testing techniques. A-State and ABI for providing the necessary facilities, and Dr. Liu from TCM Health Education and Research Center for providing the herbal compound and the project funding.
Hi, my name is Nivan.
And I'm Kaylee Rodriguez.
And along with Dr. Izalier, who is our mentor, we've been doing research with glucose sensors using the recombinant manganese peroxidase and glucose oxidase, as well as using empiriometric applications. The basis of our research stands on electrochemistry, which studies chemical reactions between charged particles and electrons, as well as the relationship between electrochemical energy and changes.
Electrochemical biosensors have proven to be useful tools for pollution detection, and this is because conventional methods such as chromatography have had various challenges, which biosensors have been able to solve. Such as a long lifetime, they're miniature analytical devices, they're sensitive enough to detect small changes in concentration, they're selective enough to detect the target, they're portable, they provide real-time measurement, and they have environmental capability. Electrochemical biosensors are proven to be useful tools to clinical and non-clinical applications. Some of the non-clinical examples are in food quality when we're testing for nutrients, anti-nutrients, etc. Enzyme-based power sources, an example of this, are the biofuel cells, which use enzymes as catalysts, so they're using living organisms in order to produce electricity. And in forensic analysis, when we use genetic material, such as blood, urine, etc. And some clinical examples, of course, are in drugs, hormones, in our case, glucose, and these are the examples here. So a big question is why we're centering our research on glucose. And this is because diabetes mellitus is a major public health problem and the leading cause of death and disability in the US and in the world. Its diagnosis and management requires everyday monitoring of blood glucose levels, and therefore there's a need for a fast, sensitive, and reliable biosensor to detect glucose. So just as biological devices, electrochemical biosensors are becoming very, very important in industrial, environmental, and medical applications. Currently, the enzymes most commonly used in biosensors are glucose oxidase and horseradish peroxidase, but horseradish peroxidase have several disadvantages. It's highly costly, it has limited reaction conditions, and it has a low catalytic effect. Studies on manganese peroxidase have shown that it's a strong oxidizing enzyme, which shows its potential to be used in biosensors. We are producing a novel biosensor using plant-produced manganese peroxidase, which was extracted from corn, which shows the same activity as manganese peroxidase.
So in our research, we would spin coat the enzyme onto gold electrodes and also spin coat a polymer called Nafion so that it would keep the enzyme sticking to the surface of the electrode. Once we further set up biosensor, we would use empiriometry to measure the efficiency of the enzyme and Nafion on the electrode. Under experimental conditions, we kept the potential constant at 0.84 volts, and at this potential, the current that is increasing tells us that the sensor is sensitive and responsive at this potential. So as we add successive concentrations of glucose from 0.02 millimolar to 15 millimolar, we can see the change in the current. The change can be seen as a step. And in each step, such as this part that is a close-up of the graph, each step represents an addition of 20 micromolar that was added every 50 seconds. So again, the potential was kept constant, and as the concentration glucose was added, the current versus time changed. In graph B, this is a calibration curve that displays the change of the current versus the concentration, which can be seen as linear. With every change in 20 micromolars that was added, it is proportional with the current each time. And so the addition of glucose tells us there is a rapid change indicating the electrode is sensitive. In all, our research investigated a biosensor that is cost-effective, simple, sensitive, selective, and stable. It's able to respond to a wide range of glucose concentrations. The amperometric measurements of glucose that we obtained show the biosensor is successful in detecting the glucose concentrations. Additionally, we studied 10% of diet green tea with citrus, which was used to study the selectivity of the electrode. The samples and conditions we applied show the biosensor is able to respond to excellent selectivity toward glucose monitoring in this range. Our research with the novel extracted recombinant enzyme from corn is being revealed for publication right now. And with our study, the findings show the enzyme is an excellent alternative that's able to detect glucose and can easily compete with conventional enzymes such as horseradish peroxidase. We would like to thank the Arkansas Biosciences Institute for providing the fund for our research. And we also acknowledge the support from the Department of Chemistry and Physics at A-State.
We'd also like to acknowledge our mentor, Dr. Zadir. Thank you.
Thank you.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello, I'm Kern Bounds. Today, I'm going to present our research on discovery of thyroid hormone-associated non-coding ribonucleic acids in fibrotic hearts. Here is our poster. For ease of understanding, I'll break down the poster into its major components. Cardiovascular disease is the leading cause of death in Arkansas, as well as the entire United States. A large majority of cardiovascular diseases are associated with impairment in heart function, which is a strong predictor of heart failure. Heart failure is characterized by insufficient output of blood from the heart. More than half of all heart failure patients develop heart failure with preserved ejection fraction or HFpEF, and no specific treatment is available for this life-threatening condition. HFpEF present with several associated anomalies, including hypothyroidism, diabetes, obesity, hypertension, renal dysfunction, cardiac fibrosis, and many more. Hypothyroidism is a condition wherein cells do not have access to sufficient thyroid hormones and have been associated with various cardiovascular diseases, including HFpEF. Stiffening of heart structure is attributed to cardiac fibrosis due to production of excessive scar tissues. This restricts the heart's ability to fill up with blood between every ejection. Only 2% of the entire genomic DNA codes for proteins, and 85% of the genome is transcribable. The majority of the genome codes for non-coding transcripts and long non-coding RNAs, also known as lincRNAs, which comprise 70% of the long non-coding RNA pool. These lincRNAs are over 200 nucleotides long and are found to regulate physiological and pathological functions. However, there is paucity in the understanding of hypothyroidism and fibrosis-associated lincRNAs in relation to HFpEF. We studied HFpEF using one of the best available models called ZSF1. These rats present with genetic mutations in leptin receptors and displayed clinical phenotypes as seen in the human population. Preliminary data from Dr. Raj's lab using microarray technology showed significant alteration of several lincRNAs in left ventricular tissues of ZSF1 obese rats with HFpEF compared to lean controls. We hypothesized that hypothyroidism and fibrosis-associated lincRNAs act as mediators of HFpEF. All rats were euthanized using approved IACUC protocols, followed by organ isolation. The hearts were also isolated and cultured for supporting cells called fibroblasts from wild-type rats. The overactivation and proliferation of fibroblasts causes cardiac fibrosis. The fibroblasts were treated with 10 nanograms a milliliter of transforming growth factor B, or TGF-beta, for 48 hours to induce fibrosis. Then RNA was collected and converted to complementary DNA using RT2 first-strand synthesis kit. qPCR detected expression levels of select cardiac-associated lincRNAs by amplification using commercially synthesized primers. One-way ANOVA and t-test were used for statistical analysis. We found that ZSF1 obese rats showed significantly increased total body weight and heart weight and left ventricular weight. In addition, liver, kidney, and left lung weights were also increased in ZSF1 obese rats compared to lean controls. Organ weight to tibial length ratios also showed significant increases in obese rat. These data demonstrate hypertrophied left ventricles, hepatomegaly, and potential renal dysfunction, indicating a phenotype of heart failure. Previous data from the lab partly validated this using ultrasound. The quality, number, and structure of the adult rat ventricular cardiac fibroblasts were satisfactory. To identify fibrosis-associated lincRNAs, TGF-beta treated rat fibroblasts were compared to non-treated controls. We have found two significantly reduced lincRNAs, LOC103694277 and LOC102549927. LOC102556455 was significantly upregulated in hypothyroid groups. Importantly, no published studies have been found to characterize any of these lincRNAs. Thus, our findings are expected to offer novel roles of lincRNAs not only in heart failure, but also in the broader field of biology and medicine. In conclusion, our research showed a clear association of uncharacterized non-coding RNAs in thyroid hormone and TGF signaling. This is the first report to unravel novel lincRNAs that could contribute to HFpEF associated with hypothyroidism and cardiac fibrosis. These findings offer new and potential therapeutic strategies for heart failure. Here are the references cited in the poster. And I would like to thank the Create at A-State Symposium organizers for providing me with the opportunity to present, Arkansas Biosciences Institute for my undergraduate research scholar fellowship, Dr. Vishwa Rajagopalan for mentoring me, the ABI grant to Dr. Rajagopalan, co-presenters Mr. Sankalpa Chakraborty for support with experiments and presentation, and Mr. Bryce Dickerson for the experiment. All the reviewers for their valuable comments, and Dr. Lucho and all other lab members. Thank you all.
Hi, everyone. My name is Sean Michael King, and the title of my poster is The Morphology and Identification of Leukocytes in Chelonian Species of Northeastern Arkansas. Hematology is important for studying Chelonian species because it can tell us much about how they fight infection and immune function strategies in different ways. Leukocyte function in turtles shifts depending on the environment, stressors, and reproduction. Wild turtle health is not very well studied, but by analyzing their blood and cell morphology, we can learn more about how they combat infection and the endocrine trade-offs. First, we need to identify and describe Chelonia leukocytes, summarize primary leukocyte activity, identify the different parasite present in Chelonian red blood cells, and address the impact of parasite abundance in red blood cells and what it does to cause harm to the host. We want to collect a blood sample from the caudal vein in turtles, place a drop of blood on the cover slip, and use a second cover slip to swipe the blood across and create a smear. Fix in methanol for 10 minutes. Bring fixed slides to the laboratory to be stained in James Wright stain. Stain for 25 minutes and then rinse for 1 minute with 10 dunks. Next, we'll observe stained blood smears under the Nikon 1200 microscope for white blood cells, parasites, and red blood cell abnormalities. Using 4x, 10x, 20x, 40x, and 100x magnification, we'll ID white blood cells and their morphology. We want to find parasites inside the red blood cells, define key characteristics of Chelonia leukocytes. The species observed was the Trichome scripta, the red-eared slider, the Kenosternum subrum, the mud turtle, and the Chelindra serpentina, the common snapping turtle. The hematology, in figure 4, we have the Hemogregaria species. This is an intracellular red blood cell parasite. Figure 5 is eosinophils. They have large pinkish orange granules. In figure 6, we see a basophil that has very dark staining granules. Figure 7, we see a heterophil. Figure 8 is a lymphocyte. That's a very large nucleus, very dark color. Figure 9, we see a monocyte. For each of the findings, in figure 10 was the number of eosinophils and hemogregaria in red eared sliders. Our population was 8. In figure 11, the number of heterophil to lymphocyte in red eared sliders, the population was 8. In figure 12, we have the number of heterophil lymphocyte in snapping turtles, the population was 2. And in figure 13 is the number of heterophil lymphocyte in mud turtles. Hemogregaria species infection in Trichomy scripta was identified in 6 out of 8 blood slides, so 75% infection. The ratio of eosinophil to hemogregaria suggests that eosinophil counts rose in response to endoparasite presence in red blood cells. The HL ratio in Trichomy scripta show that they were under stress from baseline blood samples taken. Heterophil activity was more prominent than lymphocyte activity when they were caught. That means that their stress could have been higher at that point in time. HL ratio for Chilindra serpentina showed the stress was a high factor for heterophil activity also. The sample size for K. subrum, the mud turtle, was only 1 individual, while we only have 2 individuals for the common snapping turtle, the C. serpentina samples. We hope to gather more samples in future analysis. More samples need to be taken in order to determine how HL, parasite, and stressors change their blood cell activity. I would like to thank the Newman-Lee Lab, Dr. Lori Newman-Lee, Jennifer Terry, Alyssa Harsheim, Grant Dawson, Arkansas Bioscience Institute, and the College of Math and Sciences. Here are some of the references that helped me in understanding the infections, not only in Chelonian, but other various different reptiles. And here's my final poster. I'd like to say though, on this poster that it was a lot of work, but I had a really lot of fun time doing this. The Hemogregaria species, it does affect the turtles, and it can be given to them by leech bites, and it can actually form inside their muscles and cause sarcosis. Thank you very much for watching this presentation. I hope you learned a lot about some Chelonian species in northeastern Arkansas, and I hope to do more research with this. Thank you very much.
Good evening, everyone. My name is Hugh Pasco, and I'll be presenting my Create at A-State poster project titled Phylogenetic Analysis of Genes Involved in the Demedicalization of Plants. So here's a brief snapshot of the poster I created, and we can go into greater detail on this PowerPoint. So for the introduction and purpose, phylogenetic analyses are a key process of understanding how gene products are related. The major use shows branchways and pathways of a sequence or species divergences from its ancestors through genetic evolution. And it is a problem that has plagued a lot of researchers of organisms with complex genomes ever since the ability to sequence those genomes has been invented. And one can find conserved genes of a species with fully cataloged genome and compare it to those of the lesser known conserved regions of another species. So in this study, we use Arabidopsis thaliana demethylase proteins to find a suitable family member in our target target species, Glycine max, which is soybean. The type of phylogenetic trees was neighbor joining and maximum parsimony, which we'll go into further detail later. The objectives were looking for a specific type of demethylase protein family, which is the DML homologs, Demeter-like homologs to be more specific. And we found ROS1 and DME, which are within that family, and they are conserved in soybean. However, these genes appear to have been duplicated within the genome. And by comparison of these homologs throughout Bidder plants, we can establish connections between the well-documented genes, which are in Arabidopsis, to our soybean. Defining this study will help us understand the regulation of DNA methylation process in soybean and thereby manipulate it to make soybean more tolerant to stress conditions. We can use this study to study stress conditions, abiotic and biotic stress, but we are mostly looking at P. sojei, which is a pathogen that plagues soybeans and can really cause harm in your crop yield. Here are two figures. The figure on the left shows an alignment process of the sequences of these genes, and this shows their domains for the Arabidopsis, which are AT down here, the ones we're looking at. So we can compare these conserved regions to the ones of a bunch of all different types of plants to find if there's any that align well, and we can use to see if they do align well. We use this to use the phylogenetic tree to see how the software groups them in families. For the methods, we used initial assortment of genes was from a pre-existing tree called transcriptional activator Demeter, which is about 200 genes from about 40 organisms. And all these gene IDs were aligned within MegaX, and a table was also constructed to keep track. Once aligned, all the major gaps needed to be cut, which is a long process of finding every single gap that might cause an error in your phylogenetic tree and cutting those out and finding a bunch of genes that might just mess up your data entirely. So you would need to cut those out. And after that, the trees were ready to be made. The neighbor joining method and the maximum parsimony were the most useful. And here is a slide showing the gene expression of DML genes in 10 near isogenic resistant lines of soybean. So this shows all these genes in their resistant lines. The discussion in action, we have the whole, these highly studied genomes can be used for reference for the homologs and species that are well done. We have studied that, and we can go into detail of the trees that we use. So for maximum parsimony, it uses an optimality criterion to minimize the aspect of homoplasy. This means, in short, that maximum parsi tree is the shortest possible tree that explains the data the best, like an Occam's razor. So it takes all these different factors and kind of cuts them away. A neighbor joining tree uses algorithm based on a Q-metrics, which is a mathematical algorithm, and is a fast, practical way to analyze a large data set while also using a bootstrap, which shows the accuracy of those nodes that we saw earlier. And we can combine these two as they work. They have both high points and they both cover each other's low points. So that's why we use these two different trees. And for my acknowledgements, I'd like to thank these people. They helped me a lot, and I couldn't have done it without them. And here were my references for the project. Thank you so much.
I'm presenting my project on how the cytoskeletal protein CAP1 fulfills context-dependent functions in adhesion and migration of colon cancer cells. The 5-year survival rate of colon cancer patient changes from 90% at the localized stage to only 14% with distant metastasis. Insight into the mechanism of metastatic progression, such as the involvement of CAP1 in cell adhesion and migration, may lead to strategies suppressing the invasive cycle of the disease. Our hypothesis is that CAP1 fulfills context-dependent functions in regulating cell adhesion and migration of colon cancer cells, and FAC and COFLIN are at least partially responsible for the underlying mechanisms of this function of CAP1. In Figure 1, we found no evidence supporting the upregulation of CAP1 in colon cancer cells, as high-throughput screening results suggested. Also, platelet-derived growth factor, PGF, is known to stimulate the actin cytoskeleton rearrangement and cell migration. In both SW480 and HCT116 colon cancer cells, PGF induced CAP1 dephosphorylation, suggesting CAP1 may be partially responsible for this stimulation by PGF, since CAP1 is thought to be active in its phosphorylated form. Next, in Figure 2, CAP1 knockdown clones, along with vector control cells, were generated through shRNA transfection in 2 colon cancer cell lines, HCT116 and SW480 from early and late stage localized cancer, respectively. The clones with a star represents those with sufficient CAP1 depletion to be used in marker testing and functional assays. Cofulin is important for cell migration and is active in its dephosphorylated form. Through Western blotting, we found no effect in cofulin activity for SW480 CAP1 knockdown clones, while HCT116 CAP1 knockdown clones had increased cofulin activity. To detect the effects of CAP1 knockdown on cell migration, wound healing assays were performed on both SW480 and HCT116 vector and CAP1 knockdown clones. CAP1 knockdown increased cell migration in both SW480 and HCT116, with HCT116 having the most significant increase. This may correlate with the activated cofulin in HCT116. In Figure 3, to detect the effects of CAP1 knockdown on the actin cytoskeleton in colon cancer cells, SW480 clones were stained with fluorine to look at the actin stress fibers. CAP1 knockdown increased the number of actin stress fibers in SW480, as well increased the cell size and the number of multinucleated cells. In HCT116, CAP1 knockdown led to increased cell size and enhanced lamnipodia formation. Next, in Figure 4, we see that FAC is a signaling protein important for regulating cell adhesion that is active in its phosphorylated form. Through Western blotting, it was found that CAP1 knockdown reduced activity in SW480 and increased FAC activity in HCT116. Another way to detect the effect of CAP1 knockdown on cell adhesion was through cell adhesion assays. The results correlate with the FAC results, with SW480 CAP1 knockdown clones having reduced adhesion and HCT116 CAP1 knockdown clones having increased adhesion. In Figure 5, as previously stated, CAP1 is thought to be more active in its dephosphorylated form. PKC, protein kinase C, induced CAP1 dephosphorylation through treatment with PMA. We wanted to confirm this in colon cancer cells. In both SW4 and HCT116, CAP1 was induced at 2 hours for SW480 and 1 hour for HCT116. PKC activation led to increased adhesion in SW480 and reduced adhesion in HCT116, which are opposite of the effects of CAP1 knockdown. This supports the CAP1 function in cell adhesion of colon cancer cells, and PKC may be another underlying mechanism through this function. Lastly, the small GTPase RAP1 that is involved in many biological responses, including adhesion, was more strongly detected in 2 colon cancer cell lines when compared to HeLa and MCF7. That was HCT116, SW620. RAP1 activation assays were performed to detect the changes in the activity of the 2 colon cancer cells of this project's focus, HCT116 and SW480. In SW480, RAP activity reduced, while in HCT116, RAP activity increased.
Hello, my name is Mary Amber Beach, and I'm an undergraduate in the Newman Lee Lab, and my project is on the relationship between body mass, follicular size, and egg production in red-eared sliders. Red-eared sliders are a multi-clutching species. This means that they produce multiple clutches of eggs simultaneously. So you can see in Figure 1, they have 1 clutch of eggs that's ready to be laid. The second clutch is follicles that are going to be shelled soon. And then the third clutch is earlier stage follicles. As those females make the ovum, they use reproductive effort, and that reproductive effort goes into the egg development, and which influences the overall body mass, as shown in Figure 2. So we can see that body mass can be indicative of reproductive effort. Also, going along with the follicles, there is this degeneration of follicles known as follicular atresia. And I also looked at these atretic follicles in my project. So my research goals was to develop a methodology to consistently quantify the reproductive effort of these turtles using these ultrasounds. And I also wanted to determine if the mass of the turtle had a relationship to the size and quantity of each ovum. So my hypothesis is that I think that larger turtles are going to have a higher overall mass compared to females with smaller follicles and eggs. And also they're going to have a higher quantity as well. So for the field, the first thing that we did was we would trap the turtles. We would trap them using box traps, hoop nets, and opportunistic sampling, as well as the large fight net shown in figure 4. And figure 3 is an opportunistic sampling that we found on the side of the road. Then we would use this portable ultrasound to conduct the ultrasound. And what we would do was use water as a medium. We would gently caress the turtle's leg out of its shell, use that probe and do a ultrasound of the inguinal area on each side of the turtle. And that can be seen in figure 5. And all these animals were released within 12 hours of capture. To actually analyze the ultrasound, I used the protocol with ImageJ, which is a software. And the first thing I would do is set the scale for it to be able to get an accurate measure. Then I would analyze the ultrasound for 10 minutes and mark it with the ellipse tool. And I would record the number that was assigned to each ellipse in a master spreadsheet. And then I would take the perpendicular measurements of the ova and calculate the average diameter. So for ovum ID, you can see the first one is atritic follicles. Those are the really, really small ones. So they're striated white ellipses and they're variable in size. And there are some on top of the egg, the large egg that can be seen in the middle of the second ultrasound. The follicles are white grayish ellipses and they have clear boundaries, and they generally occur adjacent to other follicles because they're in the oviduct. The eggs are the large ellipses that are characterized by a thick white band, and you can see the largest ova on there is the egg, and there's also a grayish yolk in the middle. Below that is the taskbar for ImageJ and 3 tools that were mainly used for those. And that's the line tool, the ellipse tool, and the zoom tool, just so I could get a better look at each ultrasound. Work moving forward, I want to use this methodology to analyze the ultrasound data from 2019-2020, and then use this protocol for future sampling. And I want to compare the quantitative ultrasound data with recorded mass and be able to apply that to data analyses involving body condition. I'd like to thank Christian Vickery, Taylor Fedor, Emily Field, Alyssa Hartzheim for their assistance in the field, along with other volunteers who joined us in the field. And this project is funded by the A-State Department of Biological Sciences, Arkansas Biosciences Institute, Arkansas Audubon Society, and SSAR Grants in Herpetology. Thank you.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hi, my name is Amelia Atwell, and I'd like to thank you for listening to my talk on using subwatersheds to determine variations in water quality within the Cache River Drainage Basin in eastern Arkansas. Nearly 50% of all streams in the United States are listed as impaired for not meeting their designated use. The leading cause of these impairments is non-point source pollution, and agriculture serves as the #1 source of non-point source pollution into streams of the United States. These pollutants can range from pathogens from cattle and pastureland to nutrients and sediment pollutants in row crop agriculture. In the state of Arkansas, agriculture makes up a little more than 1/3 of all land use, at 36%. 21% of that is used for row crops, denoted by red in the picture, and the remaining 15% is pasture land. Both of these types of agriculture have led to over 1,600 kilometers of streams being listed as impaired in the state of Arkansas. Looking at the Cache River watershed, which is located in northeast Arkansas, where I'm doing my research, it is located in the Mississippi Alluvial Plain, which is better known as the Delta ecoregion. And the Mississippi Alluvial Plain is a highly diverse ecoregion. It is also a tributary to the White River and is part of the greater Mississippi River Basin. The cache is also highly important. It's home to a variety of animal species and serves as a large economic importance, as 8 of the top 18 rice-growing counties in the entire United States are incorporated within it, and it's also part of the Mississippi Flyway, which has a lot of duck hunters coming in. The cache is naturally divided by ecoregions, with 87% of the subwatershed falling within the Mississippi alluvial plain, while the remaining 13% is on Crawley's Ridge. And so overall, the watershed is 70% agriculture, with 67% of that being row crops. And as you can see from the figure, the land use is vastly different between the two ecoregions. In the Mississippi alluvial plain, 80% of the land is used for row crops, with rice and soybeans being the most prevalent. There's only about 1% of the forested land within this portion of the watershed. Crawley's Ridge, however, is over 50% forested and only about 15% row crop agriculture. This is also where the majority of the urban areas are within the watershed. So, the objective of my study is to compare tributaries from the Mississippi alluvial plain and Crawley's Ridge ecoregion to determine if water quality differences exist between tributaries. My dissertation research is looking at 12 tributaries of the Upper Cache River watershed, so I selected 2 for comparison based on contrasting ecoregion and of similar sizes. So I settled on West Cache River Ditch and Scatter Creek subwatersheds. West Cache River Ditch is located entirely within the Mississippi alluvial plain and is 92% row crop agriculture. There are very limited, if any, stream buffers along the stream channels in this particular watershed. Scatter Creek is almost entirely on Crawley's Ridge, with only about 1.3% within Mississippi alluvial plain. 66% of the land use is forested, with only 12% being row crop agriculture, which is within the Mississippi alluvial plain portion. Stream buffers are limited within the Mississippi alluvial plain portion, but the rest of the watershed, the buffers are often much thicker. So I went out once a week from October 2017 to September 2020 and did physicochemical measurements where I looked at pH, conductivity, temperature, and dissolved oxygen once a week. I also brought back a sample of water in which I measured for turbidity, which is an indicator of sedimentation. And I chose these parameters because they all have state standards and/or are used to help monitor water quality changes that may negatively impact aquatic organisms. Looking at the results, the West Cache River Ditch was higher in all parameters except for dissolved oxygen. And if you know the relationship between dissolved oxygen and water temperature, this makes sense because warmer waters hold less dissolved oxygen. The West Cache River Ditch was also 2.5 times higher in conductivity and 8 times higher in turbidity. And while the underlying geology may play a factor in the 2.5 times difference in conductivity is more likely affected by land use, as conductivity looks at the amount of salts in the water, and the heavy use of fertilizers may increase conductivity within a stream. Previous studies have also shown that forested streams often have 30% less sediment deposition than those of agricultural lands, and the 8 times higher in turbidity of West Cache River Ditch and Scatter Creek may not be 30% difference. It does show the general trend. So the importance of this work shows that by comparing streams of the two ecoregions, we can see the effect that high agricultural intensity and limited stream buffers can play on streams. And because stream pollution is so prevalent throughout the world, it's important that we work to protect them. And while this study is only on 2 small tributaries in the Cache River, the implications of the importance of stream buffers can be applied elsewhere. And with that, I would like to thank my committee, my funding sources, field assistants, Ecotox employees, and you for listening to my presentation. Thank you.
Hi everyone, my name is Caitlin DeVellis and I'm an MS biology student. And today I'll be presenting to you a portion of my thesis project, the relationship between North American birds and Bergmann's rule in the context of climate change. First, there are 3 main components. The first one is Bergmann's rule, which is a biogeographic rule, which explains surface area to volume ratios in relation to climate, where in warmer climates, you're going to have smaller bodied individuals, and in cooler climates, you're going to have larger bodied individuals. Secondly is climate change, which is affecting all of Earth's ecosystems and has increased the frequency and intensity of natural disasters worldwide. And we've been observing some consequences for birds, particularly with their spatial, phenological, behavioral, and morphological shifts. Then a third portion, which I won't be discussing today, are life history and other traits. The significance of this project are that bird populations and bird body size have been declined since 1970. So if we can get a better understanding of these trends and why they are happening, then we can have better conservation initiatives that are more informed. For my research objectives, I first want to identify a change in body size of North American birds, and then I would like to identify if these trends follow Bergmann's rule. I hypothesize that Bergmann's rule does apply temporally to North American birds in the context of climate change, and then I predict that body size will show a decrease over the past several decades. So for my methods, we first contacted the bird banding lab to identify species that we want to include in this project. We then contacted the Monitoring Avian Productivity and Survivorship Program to get some initial data and to get a better idea of who would like to contribute to this project, which led to our outreach, where you can see on the map on the right, we got 72 contributors outside of the MAPS program. So I have 23 study species overall. However, we'll be focusing on these 3 today. So for data analysis, we use the program R. We've separated the data based on season. We then performed a spatial trend based on latitudinal and elevational position. We use linear mixed models with year as a random effect to control for temporal autocorrelation. And then lastly, we use the information theoretic approach and AIC to determine the best models using the principle of parsimony when applicable. So for my preliminary results for American kestrels, this is again the first objective. We noticed that there were no trends for any season besides the post-breeding migration, where we saw a decline in both mass and wing chord. This was the same trend for Cooper's hawks. Again, no trend except for the post-breeding migration, where we saw a decline in both morphometrics. And then lastly, for sharp-shinned hawks, we saw no trend for the breeding season. No trend for mass, though a decrease in wing chord for post-breeding migration. No trend for non-breeding season, and then an increase in mass and a decrease in wing chord for pre-breeding migration. So of these trends, only the trends for kestrels and Cooper's hawks applied to Bergmann's rule. So this was an interesting skew of results. Lack of trends maybe because of the relatively small sample sizes for seasons, as you can see, that's noted below. For the kestrels and Cooper's hawks, we saw a decrease in both morphometrics, which may indicate morphological correlation. However, it also may indicate that there are poor conditions in the migratory stopover sites and breeding grounds, or it may just be a result of sampling. And then lastly, for sharp-shinned hawks, we saw that separation between morphometric correlation, which aligns with a previous study. However, it is interesting, and I'd like to get more into that. So for future directions, I'd like to analyze the rest of my study species, and I'd also like to incorporate life history and other traits, as well as climatic variables, to see if these variables would indicate, better indicate morphological changes over time. Lastly, I'd like to acknowledge my committee members, Dr. Virginia Rowland, Dr. Thom Bovis, and Dr. Travis Marsico, as well as A-State and all of my financial contributors to allow me to do this research, as well as all of my data contributors, some of which are listed below. Lastly, if you have any questions, feel free to email me at caitlin.debelis@sml.astate.edu. I'll be happy to answer your questions.
Hello everyone, my name is Gaurav Gojral, a second-year master's student here in Arkansas State University in Molecular Biosciences graduate program. I have been working under supervision of Dr. Fabricio Medina Bolivar on this particular project. The title of my project is Assessing the Role of Crenulated Spinoids on Abiotic Stress in Peanut. Peanut plant has a huge economic importance due to its benefit to human health as well as its consumption need. Let me briefly talk about life cycle of a peanut plant. Seeds of peanut plant when germinated give rise to young seedlings, which upon maturity produces flowers. Then these flowers then enters the soil and give rise to peanut under the soil. During growth and development of peanut plant, it faces a particular stress called as drought, which causes death of the plant, and this causes an annual loss of around $520 million during production of peanut plant worldwide. Interestingly, in response to this stress, peanut plant activates its defense mechanism, which causes production of these natural compounds to fight against the stress. And in our lab, we have developed a system called as peanut hair root system to produce these compounds in a large amount. And the hair roots are produced by infecting the leaf of peanut plant with a special type of bacteria called as Actobacterium rhizogenus, which causes production of roots on leaf of the plant. And these roots can be grown in lab conditions in flask with media. And these roots, when stressed with certain group of chemicals called as elicitors, produces change in color of the media, resulting in production of stilbenoids and their derivatives. And there are certain genes which are responsible for production of these compounds. So in our lab, we have proposed a pathway for production of these stilbenoids and their derivatives called as prenylated stilbenoids. And there are particularly two genes which are responsible for production of these prenylated stilbenoids, and those are ASR4DT1 and ASR3'DT1. So for this project, we have used a molecular technique called as RNAi to silence the expression of one particular gene called as ASR4DT1, and we'll be observing the change in level of stilbenoids after silencing the expression of these genes and studying the role of the prenylated stilbenoids against abiotic stress. So the overall hypothesis of this project is that peanut hair root produces selected prenylated stilbenoids with potential antioxidant capacity upon exposure to oxidative stress. And to test this hypothesis, we will be developing transgenic hair roots harboring RNA construct to manipulate the class and level of prenylated stilbenoids. And we'll be analyzing the transgenic hair roots for stilbenoid prenyltransferase gene expression, stilbenoids, and antioxidant capacity. In this project, so far we have been able to establish two transgenic hair roots harboring the RNA construct, namely Georgia-Green RNA2 and Georgia-Green RNA5, and one wild-type hair root. And these hair roots were then elicited using elicitors for 168 hours, and we could see color change in the media after the elicited treatment. So wild-type hair root had the normal yellow color after the elicited treatment, which was obvious. But interestingly, in the RNA hair root, the media color changed to brown after elicited treatment. So this made us curious, and then we used analytical technique called as SPLC to observe what was actually in the media, and then what we found was pretty interesting. In the RNA hair roots, which is the chromatogram of the above two figures, so that there was only resveratrol production in RNA hair roots as a major dominant peak, and there was no detectable level of other prenylated stilbenoids. Whereas in control hair roots, we can see normal expression profile with resveratrol and other prenylated stilbenoids. The summary of this research till now is that transgenic hair roots with prenyltransferase gene knockdown were established, and RNA approach targeting a stilbenoid-specific prenyltransferase led to non-detectable level of prenylated stilbenoids in elicited hair roots. And further work focuses on validating the targeted prenyltransferase gene knockdown using RT-qPCR, which is again a molecular technique, and evaluating the role of these hair roots against with different drought-focused assays to assess the role of prenylated stilbenoids on abiotic stress tolerance. And this brings me to end of my presentation, and I'd like to thank the entire Medina Bolivar Laboratory for their help and support. Thank you.
Hello, my name is Alyssa Hartzheim, and I'm a graduate student in the Newman Lee Lab. And I would like to present my project, Optimization and Implications of Microbial Cytokilling Assays for 2 threatened species of tortoise under zoo conditions. Microbial cytokilling assays are often used to assess the immunocompetence of an individual using known amounts of serum and microbe. Serum in this instance is achieved by taking a blood sample, centrifuging it, and then removing the clot. For this assay, the serum can either be stored frozen and then thawed, and then this would assess immune components such as the complement system, natural antibodies, and antimicrobial peptides. Or the serum can be used live, and this assesses immune components such as phagocytic cells, natural killer cells, the complement system, natural antibodies, and antimicrobial peptides. For my project, I validated 3 different microbes, Staphylococcus aureus, Escherichia coli, and Salmonella enterica. Now, the optimization process is often needed because immune function cannot be universally assessed. Instead, species-specific methods are needed. Not only may microbial cytoability differ between species, but it may differ between microbes as well. The goal of this optimization was to achieve 50% total killing of the microbe by the serum. To do this, serum volume was varied in a stepwise manner from 3 to 15 microliters to determine the ideal volume. Microbe volume was kept the same for all assays at 6 microliters. The study species for this project were the Indian star tortoise and the northern spider tortoise. Both study populations for these species are currently housed at the Memphis Zoo in their herpetological collection. Now the results. Both species were able to mount a response against all microbes. Both species had adequate killing against S. aureus using frozen serum. However, both species had no measurable response for both E. coli and S. enterica when using frozen serum. So, to account for this, live serum was then used. When looking at the results from S. aureus, both species required the same volume of serum at 6.5 microliters, and this achieved 20 to 30% killing of the microbe. For E. coli, a much lower volume of serum was required for both species, both of them requiring only 3 microliters. However, 70 to 80% killing of the microbe was achieved using this volume, but keep in mind this was done using live serum. For S. enterica, this is where we notice a difference between the species. The Indian star tortoise required only 5 microliters of serum, and this accounted for 40% of the killing, whereas the spider tortoises needed almost twice that at 11 microliters of serum, and this ended up resulting in 65% killing of the microbe. Again, this was done using live serum. Now, some explanations and future analysis. Both species exhibited substantial killing ability against S. aureus when using frozen serum. This may be because these tortoises may rely preferentially on the complement system, natural antibodies, and antimicrobial peptides when mounting a response against S. aureus instead of phagocytic cells or natural killer cells. Also, it may be possible that this microbe is more frequently encountered in this tortoise's environment, and these tortoise species may have subsequently evolved a stronger immune response to account for this. Now, when looking at E. coli and S. enterica, both species only achieved adequate killing when using live serum and not frozen. The freezing process destroys phagocytic cells. These tortoises may rely primarily on these phagocytic cells and natural killer cells when mounting an immune response against these microbes. This may explain the lack of killing that was observed when using only frozen serum. I would like to acknowledge my advisor, Dr. Lori Newman-Lee, and I would like to acknowledge Jen Terry, Dr. Sheila Pu, Ali Bogish, and Mary Ella Khan for their assistance in sample collection over these many months. And I would like to thank the Memphis Zookeepers and staff for their assistance. And I would like to thank you, audience, for sitting and listening to my presentation.
Hi, I'm Roki Hassan, a PhD candidate here at Arkansas State University, and my advisor is Dr. Fabricio Medina Bolivar. Today, I'm presenting our research on finding the potential cardioprotective function of the peanut hair root extract and arachidin. Despite great therapeutic advances, cardiovascular disease remains a major health problem and the leading cause of mortality here in USA. Arkansas ranks 3rd among all the states in mortality rate. To further improve the symptoms and survival of CVD patients, scientists are consistently looking for novel therapeutic strategies. Resveratrol is one of the most well-studied stilbene, which initially found in red wine and later also found in peanut, showed a wide range of cardiac health benefit against coronary artery disease, hypertension, atherosclerosis, and oxidative stress. However, because of faster metabolism, it has limited bioavailability. Pre-19 stilbene arachidins are derivative of resveratrol found in illicito-treated peanut hair roots. Collaborative research from our lab found that because of the prenyl group marked in red circles, arachidins are potentially more bioavailable. Some literature is suggesting cardioprotective effect of arachidins. However, the exact mechanism of cardioprotective function of arachidin yet to discover. Our lab uses peanut hair root system to produce these arachidins. In this project, we will elicit, extract, and purify the arachidins from peanut hair root medium and check its cardioprotective function in heart-derived cells as well as rat models. So our overall goal is to dissect the potential cardioprotective role of the arachidine, and the hypothesis is that peanut hair roots enriched in arachidine and purified arachidines exhibit cardioprotective properties in vitro and in vivo. To test the hypothesis, we have listed 3 objectives in this project. The experimental workflow for the first 2 aim is here. After elucidation and extraction of the peanut hair root culture medium, arachidines was purified using HPLC method, which later has been used to treat the cells. We have used the HPLC method to purify the arachidines from peanut hair root culture medium after elucidation. Cytotoxicity of the extract and arachidines were checked in H9C2 cardiomyocyte cells using real-time glow empty cell viability assay in various time points. We have checked the optimum non-toxic concentration for the cardiac cells in 4, 24 and 48 hours, and here the IC50 data shows in the table. For extract, it shows around 11 microgram per ml, while for both purified arachidines, it showed around 10 micromolar concentration. Later, we have treated the cells with a different concentration of hydrogen peroxide to induce oxidative stress. Here is the cell viability data against the oxidative stress. For further treatment, we have selected the 250 micromolar concentration of hydrogen peroxide for 4 hours, which kills almost 50% of the cardiac cells due to oxidative stress. Later, we wanted to see whether our extract and purified arachidine can protect the cells against the hydrogen peroxide mediated oxidative stress, and that's why we pretreated with the arachidines or extract for 2 hours before introducing the hydrogen peroxide. Here is the result that showed that both the arachidine 3 and extract can protect the cells against hydrogen peroxide mediated oxidative stress in 10 micromolar and 10 microgram per ml concentration respectively. For the third aim, we did a 6-week long pilot project with rats where we had 4 different groups. Control treated with PTU, which creates the cardiac injury, 40 and 200 mg per kg per day extract treated along with PTU. We have collected morphometric, blood and echocardiogram data, and after the experiment, heart tissues, which has been used for further analysis. Here are the body weight data of each group before and after the experiment, and we have used tibial length to normalize. After the 6-week experiment, we found that the extract, which is here on green color, 40 mg per kg per day, and this one is 200 mg per kg per day, does not affect the rat body weight significantly. Later, we did RT-qPCR with the heart tissues to check various cardiac markers, and here the qPCR result for 2 important cardiac markers, myosin alpha and myosin beta. We found that the 40 mg per kg per day concentration recovers the myosin level compared to the PTU-induced cardiac injury group. In conclusion, we found that both the extract and purified arachidine can protect the cardiac cells against oxidative stress, and our pilot study data is suggesting that 40 mg per kg per day extract treatment can help the PTU-mediated cardiac injury in rats. We are conducting further animal study and various molecular techniques to increase our understanding. Thank you for giving me the opportunity to present my research. Also, I want to thank everyone involved in this project and the funding agencies, especially the Arkansas Bioscience Institute. Thank you.
Hello everyone, I'm Uddhav Karki, a PhD student in Molecular Bioscience graduate program at Arkansas State University. I am working under supervision of Dr. Jesu. Today I am going to present one of my PhD dissertation project. In this project, I am trying to produce the functional human erythropoietin in tobacco cells for large-scale ex vivo generation of the red blood cells. Let's start with the research background. Blood is a crucial component of our body, and we need to have the additional supply of blood during different complications, especially in the accident. The blood is only produced inside the body, so blood donation is one and only option. In this global COVID-19 pandemic, the blood supply was impacted because it was not easy to organize the blood donation program, as well as all people were not eligible to donate the blood. So the only solution for this problem is to produce the human blood cells outside of the body. This is the long-term goal of this project, and I am working as one part of this project to produce the human erythropoietin in the plant cells. This human erythropoietin is a key growth regulators for the proliferation and differentiation of the hematopoietic stem cells into the red blood cells. So the overall goal of this project is to higher level of production of human erythropoietin in the plant cell culture system by using some special tag called HIPCP tag. Erythropoietin is a important biotherapeutic protein which has the diverse clinical applications. They are used for treatment of the chronic renal failure, anemia, different cancers, and anemia associated with the HIV/AIDS. These kind of the biotherapeutic proteins are produced in the different biological production platforms. Among them, the bacteria and the mammalian cell culture system are the major one. But still, there are some challenges in animal cell culture system because they are costly, time-consuming, and there are more chance of cross-contamination. And bacterial cells cannot produce the more complex humanized proteins. So to overcome these problems, plant can be the best alternative production platforms because it was already approved that plant-derived therapeutic proteins, glucocerebrosidase was approved by the FDA in 2012. But still, there are some limitations for protein biotherapeutics. They have the instability and the shorter serum half-life. So to overcome these problems, we have the special technology called HIPCP technology. By using this technology, we use the serine and proline tag in the C-terminal and N-terminal region of the erythropoietin, which helps with secretion, solubility, and stability of the protein. Let's start with the research plan. This is a schematic overview of the research plan, and I already cloned the human erythropoietin gene with a serine and proline 20 repeats tag, and this gene is transformed into the tobacco-derived suspension culture cells called BY2 cells, and these BY2 cells produce erythropoietin and secrete the erythropoietin into the media. I harvested the media and purified the erythropoietin by using the different protein purification approach from the ECTA start, and the purified protein was tested its biological function by using the hematopoietic stem cells function and differentiation into the red blood cells. Let's discuss about the results. We already established the stable lines having the higher level expression of the SP20 tagged the erythropoietin in the tobacco BY2 cells. We also found that the SP20 tagged erythropoietin has a higher level of secretion as compared to the non-tagged one. I found that it is about 18.5-fold more secretion as compared to the non-tagged one. And we also optimized the optimum time for the harvesting of the media. It is 12 to 14 days. And we also established the expansion and differentiation of the hematopoietic stem cells by using the commercial cytokines, erythropoietin, interleukin 3. These are the growth factors. We use the flow cytometer to see the surface marker of the cells. Initially, the hematopoietic stem cells had the CD34 surface marker. It has changed into the CD71 after 14 days of the differentiation and expansion. So this is a summary, and till now we have the good lines of the BY2 cells with these proteins, and we have the purified this tagged erythropoietin from the plant cells, and we already established the differentiation of the expansion differentiation of the hematopoietin cells. And in future, we will characterize the molecular structure of this erythropoietin and check its biological function in vitro. I'm thankful to my advisor, Dr. Su, my committee members, lab mates, Arkansas State University, Molecular Bioscience Department, ABI, NSF, and Arkansas State for the funding support. Thank you so much for listening to me. Thank you.
Hello everybody, my name is Sharon Podial and I'm a master's student in the Department of Molecular Biosciences here at Arkansas State University. I look forward to talking to you today about my project titled Differential Methylation between Moth and Infected Soybean Seedling. Did you know that the United Nations has estimated the world's population will be 9.7 billion by 2050 and that food demand will grow massively by 58 to 19%? To address all these problems and boost food security for the future, many researchers from around the world are trying to find a way to improve sustainable, high-nutrient crop production. In our laboratory, we work with soybean, the second largest crop in the US, with annual sales of $39 billion, reported in 2018. Soybean has gained popularity due to its high nutritional value, its flexibility in different industries, and much more. However, A pathogen called Phytophthora soozae is one of the primary soybean pathogen that causes Phytophthora stem and root rot, and annual crop losses of up to $200 million in the United States alone. Researchers and farmers use P. soozae resistant gene (RPS) genes for primary disease control as no fungicides are available. RPS protein released by the RPS gene allows the plant to recognize a virulent or AVI protein produced by Phytophthora soozae in the cell. This interaction will activate the downstream regulatory and defense-related genes and provide plant resistance. Pathogens, though they are clever, they alter the composition of their AVI protein such that the plant does not know it and ultimately destroys the plant. Due to this reason, these RPS genes are only useful in the field for up to 8 years. Therefore, to find a sustainable disease management strategy, it is important to consider the downstream pathway of the plant defense mechanism. One of the crucial steps is the epigenetic modification of plant DNA. In this project, I have concentrated mainly on one of its DNA methylation kind. DNA methylation is the addition of the methyl group to the fifth position of the cytosine carbon. This may have a significant effect on the expression of the gene, which will ultimately affect the production of different forms of phenotypic and genotypic proteins in the plant. Thus, the overall research question for this project is how epigenetic modifications such as DNA methylation affect the transcriptional gene regulation. To solve this problem, the aim of this project is to detect differential DNA methylation in Phytophthora soozae infected soybean using whole genome bisulfite sequencing. In my experiment, there are total 6 soybean samples. Williams 82 cultivar of soybean, 2 infected with race 1 P. soozae, 2 infected with mock, and 2 infected with race 25 P. soozae. I want to thank Dr. Robertson's lab from Iowa State University for providing these plant samples. Now, let's talk about the approach of this project. The samples were prepared for whole genome bisulfite sequencing, or WGBS. It is a gold standard high throughput sequencing technique for genome-wide methylation at single base resolution, which provides methylation pattern of all 3 methylation contexts, CpG, CSG, and CHS, and useful to compare different kind of samples treated versus mock, and can be used to predict correlation between cytosine methylation and transcriptional regulation. First, the genomic DNA is extracted, fragmented, end repair is done, and then methylated adapters are ligated on the both end of the DNA fragment. After adapter ligation, the most essential step of this library preparation, bisulfite conversion, is done, where unmethylated cytosine are converted into uracil and eventually into thiamine during the PCR, and the methylated cytosine remains same. This step is very important because it will determine the number of methylated cytosine we will get after sequencing our library. The quality of bisulfite converted DNA are then detected using bioanalyzer and sequencing using sequencer. The bioanalyzer gel report showed that the average size of library was around 250 base pair, as we predicted. That means the library can be sequenced now. After sequencing the raw data, it was for the quality assessment, multi-QC was used, and also CutAdapt to trim the adapters. Then in downstream, in future, we will be using Bismarck and MethylKit to analyze further data. The Fred box plot shows that the quality score of our data was between 30 to 40, which means the base calling was good. The per sequence GC content per percent graph was symmetrical like the control line. For the second pair end, read the proportion of complementary bases shifted because of the breeze amplification during sequencing. Even distribution of bases throughout the sequence was seen, which means the base calling isn't biased. Also, GC plot shows the proportion of adenine and thymine in large, while the guanine cytosine proportion is reduced because unmethylated cytosine are converted to thymine. This indicates bisulfite conversion has worked. Considering all the results, in future we will be using other bioinformatics tools to analyze the data and research on other bisulfite library preparation kit. At the end, I want to acknowledge my advisor and co-author Dr. Silla, our collaborator and co-author Dr. Milad, and Dr. Robertson, Dr. Clarice for the plant sample, A-State internal seed grant for funding, all of my lab mates, A-State ABI, and AVI for the infrastructures and facilities, create A-State for these presentation opportunities. Here you can scan to get the reference for my presentation and my email if you had any question. Thank you.
Hello everyone, I'm Santara Ratnayaka, Master's Biology student. I'm presenting my project on machine learning based gene regulatory network model during soybean Phytophthora zoae interactions. First, I would like to explain about the importance of the soybeans. Soybeans is the second largest crop in US with annual revenue around 40 billion, and USA is the largest soybean producer and second leading soybean exporter. And if you look at this pie chart over here, it shows the US soybean domestic consumption share by segment for year 2013 and 2014. So this gives us an idea in which areas the soybean is useful and what kind of a involvement the soybean has with US economy and household. However, there are many challenges the soybean industry faces, and one of them is the large crop losses due to pathogens. Phytophthora zoae causes Phytophthora root and stem rot in soybeans, and the approximate loss is around $200 million annually. In order to improve the resistance of soybean against the pathogens, we need to understand the soybean defense response against the pathogens. Figure A over here shows the race-specific single dominant genes acting with affluence genes of pathogens and triggering a down signaling process which causes protein modification and transcriptome reprogramming, which trigger the defense action of the plant. But when the pathogens evolve and mutate, the RPS genes fail to trigger the defense action, so resulting in a huge crop losses. Therefore, we need to dissect the downstream signaling process to understand the key factors of this defense mechanism. There we believe the main fact is to understand the downstream transcription reprogramming of the soybean and identify the key transcription factors. So we aim to build a predictive gene regulatory network model based on support vector machine approach and predict new transcription targets to improve the soybean resistance against Phytophthora zoae. In our methodology, initially, we plan to use publicly available microarray data, RNA sequence data, and make a uniform data set and build the initial model. And then we will retrain our support vector machine model using experimentally verified data from Vijayaratna lab. Then we will make new predictions based on our model. So this is the basic structure of our model. So initially we will use the pre-existing knowledge and the experimentally verified data. Then we will train our model using both negative, positive, and neutral input data. And then when we use transcription and gene pairs, we can get either negative or positive hit based on the model. So this way we can predict new transcription factor gene pairs which are important in the fight against the pathogen. And eventually we will evaluate the model based on precision, recall, and F1 score, which are based on true positive, true negative, and false positive. If you have any questions, please contact me. And I would like to thank my advisor, Dr. Asel Vijayaratna, and all the members of Vijayaratna Lab Group. Thank you.
Hello, everybody. My name is Krishna Sharma. I'm a PhD student in the Molecular Biosciences graduate program, and I work with Dr. C. Here I'm going to discuss neural regeneration in peripheral nerve, and the title of my today's presentation is Tempocellulose substratum with stem cell exosomes to facilitate sciatic nerve regeneration. Our nervous system has two components, central nervous system and peripheral nervous system. The central nerve brain and the spinal cord are the parts of central nervous system, and the nerves coming from the brain and the spinal cord form the peripheral nervous system. Peripheral nerves are the parts of neurons. As you see in this cartoon diagram, a neuron has a large cell body and a long process called axons. Axon has sheath called as myelin sheath, which is insulating sheath as in the electrical wire. When there is damage or injury to the nerves, the first thing happens is there is loss of myelin sheath called as demyelination. And with the loss of myelination, there is degeneration of neurons, and ultimately there is death of neuron. Due to death of neuron, there is loss of sensory and motor function loss. There are several causes for the peripheral nerve injury, such as systemic disease and localized damage. However, there is no successful treatment available for the peripheral to cure peripheral nerve injury. Recently, exosome and cellulose-based biomaterials have been considered as a potential therapeutic strategy for peripheral nerve injury. Cellulose, as you know, is a plant-based biomaterial and have been widely used in regenerative medicine. Exosomes are tiny vesicles released by active cells in the extracellular environment, and they contain proteins, lipid, RNA, and DNA, and when taken up by another cells, these exosomes release their content and influence the cellular functions. So exchange of exosomes is a way of communication between cells. So our objectives are to assess the efficacy of cellulose biomaterials and exosomes in neural regeneration in peripheral nerve injury, and also to evaluate functional and histological recovery after nerve injury. Our collaborators at UMS and University of Arkansas at Fayetteville, they converted cellulose into more stable form called tempocellulose, and from the tempocellulose they made thin film called tempo film biomaterials for the transplantation experiment. We in our lab cultured mesenchymal stem cells. The growing mesenchymal stem cells release exosomes in the culture medium. So we collected culture medium and then harvested exosomes from the culture medium using exosome isolation kit, and then characterizes those exosomes before transplanting into the animal. So to prove our hypothesis that the tempocytes and exosomes support nerve regeneration, we performed sciatic nerve cross injury, and after injury, we wrapped the injured site of the nerve with the exosome-coated tempocyte, and after surgery, transplantation surgery, we performed behavior tests at different time points to assess the sensory and motor function gain. At the end of the experiment, we collected tissue and performed immunostaining to determine nerve regeneration. So this is our behavior test. This is the results from the locomotor test, in which the animals are placed on a flat surface and allowed to freely move, and the movements of the hind limbs were observed. And based on the movement of the hind limbs, scores are given from 0 to 21. So this is the results from this locomotion test. The y-axis is representing the score, and x-axis is representing different time points at which the experiments were performed. So as we see, after surgery, all the animals improved in the BBB score. However, the film and exosome treated group performed better compared to other groups. We then performed a Rotarod test to assess the sensory motor coordination. In the Rotarod test, animals are placed on a rotating rod, which is rotating at a particular speed, and the time spent by the animals on the Rotarod is recorded. So here is the results from the Rotarod test. The y-axis is representing the time is spent on the Rotarod, and x-axis again the different time points at which the experiment was performed. Here again, we see the separation between the groups. However, the film group performed better compared to other groups. So this result shows that the exosome and cellulose supported motor function recovery and improved sensory motor coordination after sciatic nerve injury. At the end of experiment, we collected tissue and performed immunohistochemistry. And these are the fluorescence micrographs showing neurofilaments, which is a marker for the axons. And as we see in these diagrams, the treatment groups show more nerve fiber expressing neurofilament compared to the control group. So these results also shows the exosomes along with cellulose supported regeneration of sciatic nerve injury. So in summary, we can say that the mesenchymal stem cell derived exosomes along with tempo-cellulose may promote faster recovery of motor function and sensory motor coordination, and also the neural regeneration in a rat model of sciatic nerve injury. Since this is an ongoing study, we are still evaluating remyelination in the injured nerve after treatment, and also we are tracking the exosomes after transplantation, as well as we are also determining the effective concentration of exosome for neural regeneration. This study is supported by CASE, NSF, Arkansas, and the grant from NYIT to Dr. Say. Finally, I would like to thank my advisor, Dr. Say, and our lab members and our collaborators from UMS and University of Arkansas, Fayetteville. Thank you very much for listening.
Hello, my name is Jen Terry, and I'm a master's student with Dr. Lori Newman-Lee in the Biological Sciences Department. Today, I will present my poster titled Shifts in the Acute Stress Response in Female Red-Eared Sliders, Trichomis scripta. Given the finite nature of energy, energetic trade-offs must occur within an organism as shown in Figure 1. Glucocorticoids, or energy mobilizing hormones, are likely mediators of these trade-off dynamics. Thus, endocrine patterns influence innate immune function. The acute stress response can provide insight to energy dynamics occurring within individuals. During reproduction, female red-eared sliders may develop multiple clutches of eggs. This likely requires varying amounts of resources as clutches are developed simultaneously, which is depicted in Figure 2. With this in mind, I examined how bacterial killing ability and circulating corticosterone concentration following an acute stressor changes within and across months. Bacterial killing ability allows for the assessment of complement and natural antibodies, components of the innate immune system, to clear a biologically relevant pathogen like E. coli. Corticosterone is the primary glucocorticoid in reptiles like turtles. In 2019, we took a series of blood samples from red-eared sliders after we removed the animal from the net or hand captured them, which was the acute stressor. Thawed plasma was used to run bacterial killing assays and radioimmunoassays. I then conducted non-parametric Kruskal-Wallis tests and post-hoc Duns tests. Time, either month or the time of the blood sample in minutes, was used as the independent variable. I grouped samples collected in September and October due to sample size within each month. Both months are considered non-reproductive months for female red-eared sliders. Either bacterial killing ability or corticosterone concentration was used as the response variable. All captures were removed prior to analysis. As shown in Figure 5, I found that there is a significant change in bacterial killing ability between 0 and 30 minutes and 0 and 60 minutes in June. Also, bacterial killing ability at 0 minutes or baseline is lower in May and June. Notice that there is little change across time in July. Overall, bacterial killing ability is higher in August and September and October. Figure 6 shows that mean corticosterone significantly changes between 0 and 30 minutes for all months except September and October, indicating a change due to the acute stressor. Mean baseline corticosterone was higher in May compared to June and September and October. Baseline corticosterone was lower in September and October than August. These results indicate that energy is diverted toward reproduction and then is exhausted by July. This makes sense as we observed females gravid with eggs between May and July. No large follicles or eggs were detected on sonograms in August, September, or October. Thus, as reproductive activities conclude and the animal likely recovers energy through foraging, we see energy prioritized back toward innate immune function. I have provided a visual representation of this in Figure 7. Further work is required to identify nuances among individual responses, and earlier trapping in the year will capture the beginning of the reproductive period for these females. Understanding how these animals allocate their energy during obligatory processes like reproduction provides information that can be applied to questions in conservation and physiology. Thanks for checking out my poster. Supplemental materials, including the references used in this presentation, can be accessed using the QR code on the poster.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello everyone, I'm Lokesh. I'm a grad student from Biological Sciences Department working in Dr. Zhao's lab. Today I'm going to be presenting my poster, which is titled Mechanisms Underlying the Phosphorregulation of CAP1 by Protein Kinase C and Cellular Functions of the PKC epsilon isozyme in HeLa and MCF7 cells. Cancer. As we think about it, the only thing that comes to our mind is death. Over 12 million people worldwide are diagnosed with cancer every year. In 2020 itself, roughly 1.8 million people were diagnosed with different forms of cancer in the United States. These statistics tell us that how important it is to do a cancer research. Cell signaling molecules in the cells helps in normal cellular processes in our body. The dysregulation of different cell signaling molecules plays an important role in cancer development. Among several cell signaling molecules, my research focuses on an important cell signaling molecule called adenylyl cyclase associated protein, also called as CAP1, and how its phosphorregulation affects the metastasis of cancer cells. Also, my research brings other important cell signaling molecules that involves in the phosphorregulation of CAP1 and its potential role in regulating cell adhesion and migration. My hypothesis is PKC regulates CAP1 by inducing CAP1 dephosphorylation, which regulates cell adhesion and migration, potentially cell proliferation in HeLa and MCF7 cells. Figure 1 is a schematic diagram showing us an assumption of how CAP1 dephosphorylation induced by PKC through glycogen synthase kinase 3 inhibition and protein phosphatase 2A activation, which in turn regulates cellular functions like cell adhesion, migration, and proliferation in HeLa and MCF7 cells. In the figure 2, we tested the role of protein kinase C in inducing CAP1 dephosphorylation by treating the MCF7 and HeLa cells with PMA. PMA is a PKC activator. And in the figure A, the Western blot results clearly shows the dephosphorylation of CAP1 when treated with PMA. We also tested the role of PKC epsilon isozymes in inducing CAP1 dephosphorylation, and the figure B clearly shows that among different PKC isozymes of PKC epsilon plays an important role in inducing CAP1 dephosphorylation. We also tested the potential involvement of GSK3 in dephosphorylating CAP1 because it already reported that PKC inhibits GSK3. In the figure C, the Western blot results clearly shows us the increased phosphorylation of GSK3 molecule by PKC, which gives us a hint that the PKC might be going through GSK3 in dephosphorylating CAP1 by inhibiting GSK3. Because GSK3 is a kinase which can phosphorylate CAP1. So when inhibited, GSK3 cannot phosphorylate CAP1. We will also test the potential involvement of protein phosphatase 2A also called as PP2A, in phosphorylating CAP1 because the activation of PP2A by PKC is already reported. In the figure 3, the cellulation assays and cell migration assays results in HeLa and MCF7 cells clearly shows that PKC activation by PMA leads to increase in cell motility and cell attachment in both cell migration and cell adhesion assays. In the figure 4, we also tested the role of CAP1 in regulating cell adhesion and migration in HeLa and MCF7 cells by shRNA knockdown of CAP1. If you see the figure A, you can clearly see that in CAP1 knockdown cells, the attachment and cell motility in both cell adhesion and migration assays is compromised, even though the PKC is activated by PMA. In the figure B, we also tested the cell attachment and cell motility by re-expressing the CAP1 protein in HeLa cells. Surprisingly, the CAP1 re-expression restores the effect of PMA in CAP1 re-expressed RWT cells when compared to CAP1 knockdown cells. And my summary is, PKC induces CAP1 dephosphorylation when activated by PMA. The PKC epsilon is isozyme of PKC responsible for CAP1 dephosphorylation in HeLa cells. Significant increase in cell adhesion and migration is seen in MCF7 cells, but in HeLa cells, the results are not that significant. Thank you very much.
Hi, my name is Amelia Atwell, and I'd like to thank you for listening to my poster on total nutrient sediment and agricultural intensity, a 3-year study of the Cache River watershed, Arkansas. Agriculture is a leading cause of stream impairment in the United States and serves as one of the largest sources of nitrogen and phosphorus into streams. This can lead to streams that drain agricultural lands to have decreased bank stability, increased soil erosion and sedimentation, as well as excessive nutrients that can lead to eutrophication. All of these, alone or together, can lead to a reduction in water quality. My research is on the Cache River watershed, which is located in northeast Arkansas. It is a highly agricultural watershed with 8 of the top 18 rice-growing counties in the entire United States being incorporated within it. It is also listed as impaired by the Arkansas Department of Environmental Quality for excessive nutrients, turbidity, and lead. So the objective of my project was to assess sediment and nutrient contributions to the Upper Cache River watershed through weekly water quality measurements. My hypothesis is that the agricultural land intensity will have an effect on the level of sedimentations and nutrients, and I predict that low intensity subwatersheds will have the lowest levels of turbidity and nutrients over the 3-year study. Subwatershed land use intensity was determined for 12 subwatersheds using USGS National Land Cover Database satellite imagery, where I then calculated the intensity by taking the agricultural land use upstream and dividing it by the total drainage area minus non-agricultural land use downstream. I then grouped the percentages into low, moderate, high, and high intensities. Additionally, I looked at differences upstream and downstream of the main channel of the Cache River. For sediment and nutrient analysis, I went out once a week from October 2017 to September 2020 and used the bucket method to collect water. I then took a subsample of water for total nutrient analysis that was then analyzed on a scalar SAN Plus flow-through analyzer following American Public Health Association protocols. I also took a subsample back for turbidity analysis to use on a Hach turbidimeter, which also followed American Public Health Association protocols. Looking at my results, with turbidity, the land use intensities and main channel locations were similar with the exception of low and low moderate intensities. These two were significantly different from each other as well as the other 4 classifications. So over the course of 3 years, no intensity exceeded an average of 250 nephelometric turbidity units, which is a limit set by the ADEQ, but moderate high, upstream and downstream Cache River locations did exceed it close to 25% of the time. This data supports previous studies which indicate that forested lands tend to have 30% less sediment than those of agricultural lands. With total nutrient analysis, low intensity was significantly different from all other intensities in total phosphorus, while low moderate intensity was significantly different from all other intensities in both total phosphorus and total nitrogen, exceeding state standards in both. Total phosphorus state standards were exceeded for all intensities. The exceedance of both total nitrogen and total phosphorus in the low moderate intensity may have been driven by the site Big Creek Ditch, which is located in Jonesboro, because it was about 2 to 4 times higher than most other sites in both types of nutrients. When removed, total nitrogen falls within state standards for the low moderate intensity. And so the nutrients may be entering the waterway through runoff as sediment-bound particles. The exceedance and near exceedance of state standards in turbidity and total analysis and total nutrients is alarming for aquatic fauna. Having excessive turbidity and or nutrients can negatively impact stream biota because it can lead to homogenization of substrates and reduce oxygen in the waterways, ultimately reducing taxon diversity. It is important to continue to monitor the Cache River watershed and to work to reduce inputs of sediment and nutrients into the waterway. My future work will be to investigate lead contributions in the watershed and determine if lead is bioaccumulating in the tissues of green sunfish, bluegill, and mosquito fish. And with that, I would like to thank my funding sources and the Ecotox research students and staff, as well as you for listening to my presentation. Thank you.
Hello, my name is Nicholas Craddock, and I would like to thank you for listening to my poster presentation today. My research is on 4 sub-watersheds of the Buffalo National River, specifically how local pasture land usage affects each sub-watershed on a downstream gradient. It is known that pasture land can have a negative impact on aquatic ecosystems via runoff, which can carry excess fecal bacteria, nutrients, and soil. Each of these factors can negatively impact aquatic ecosystems by increasing total suspended solids, or TSS, and turbidity, decrease dissolved oxygen, cause harmful algae to bloom, and make an unsafe environment for playing in the water. The main goal of this project is to determine if contaminants are entering the Buffalo National River via these sub-watersheds. It is important because the Buffalo National River was designated as an extraordinary resource waterway by the Arkansas Department of Environmental Quality in 2008 due to its primary, secondary contact recreation, drinking water, and agricultural usage. The 4 sub-watersheds of study are Bears Outlet Creek, Brush Creek, Calf Creek, and Tomahawk Creek, each with a sample site near the headwater and outlet of each creek. Bears Outlet Creek was chosen because it has been listed as impaired in the past due to total dissolved solids. The other 3 creeks were chosen because they are in proximity to Bears Outlet Creek and flow directly into the Buffalo National River. Water quality samples are collected weekly, where I collect for pH, dissolved oxygen, or DO, conductivity, temperature, TSS, turbidity, fecal coliforms, or CFUs, and nutrient analysis. To maintain accuracy and consistency, samples are collected and analyzed following APHA and US EPA methods. Data for water quality parameters, excluding fecal coliform units, or CFUs, began in November of 2019, and CFU data began in February of 2020. All data for this presentation runs through the end of December 2020 with more to come. All calculations that include CFUs use data from February 2020 to December 2020 for uniformity, while calculations without CFUs contain data from the full dataset. The figure for CFUs does not include outliers for visual purposes. While Bear, Brush, and Calf creeks all had significant differences in pH, conductivity, and turbidity, only Bear and Brush creeks increased in pH, Bear Creek increased in conductivity, and Calf Creek increased in turbidity. Brush Creek also had a significant increase in TSS. While Brush, Calf, and Tomahawk creeks all experienced increases in CFUs, the only creek that had a significant difference was Brush Creek, and it had a decrease in CFUs. As shown in Figure 4, a Pearson's correlation matrix between all parameters showed strong relationships between turbidity, TSS, and CFUs. And that relationship was positive, while temperature and DO have a strong negative relationship. Future analysis will provide a more in-depth picture of the impact that these creeks have on the Buffalo National River main channel, as they will include land usage, nutrient analysis, and hydrologic data to explain any significant findings that are not related to pasture lands. Lastly, I would like to thank the Ecotoxicology Research Facility and personnel at Arkansas State University for helping me and providing guidance throughout this project, as well as the National Park Service for their assistance with locating most of the sample sites. And finally, thank you to the United States EPA and the Arkansas Department of Agriculture Natural Resources Division for their funding and oversight of this project.
Hi, my name is Rainey DeVroin, and I'm a master's student here at Arkansas State University, and this is my project, the water quality of subwatersheds located in the Middle White River in Arkansas. The Middle White River is located in northeast Arkansas. It flows from Ozarks, Highland to Delta ecoregions. The waterway is used year-round for commercial and recreational activities such as agriculture, industrial water supplies, and it's also one of the top fisheries in the world for trout. So why am I studying this watershed? Well, some of the sections of this watershed have been on the ADEQ's 303D list for multiple years. The 303D list is a list of the state's impaired and threatened waters. What this watershed is impaired with is turbidity, dissolved oxygen, and pathogens. The major source of this impairment is listed either agriculture or unknown. The possible unknown could be the land use around it. It's mainly forest and pasture. However, over the years, the pasture has been transformed into row crops. And by having this, it has the potential contamination of the waterway by not having any vegetation on the nearby area. So when a rain event happens, it could easily pick up that sediment and transport it to the nearest body of water. So my project objectives are to, 1, evaluate the abiotic factors of my 4 subwatersheds to assess their water quality parameters. But the problem is we do not know which subwatersheds is impaired. And so the objective is to find which subwatersheds are impaired so we're able to restore them, which then we will be able to improve the Middle White River watershed as well. I conduct this research on the 4 subwatersheds that are located in Independence County in Arkansas. There's Miller Creek, Spring Sprint Creek, Greenbrier Creek, and Lower Salado Creek. What I do is I take a field probe meter to the field weekly to collect the conductivity, pH, dissolved oxygen, and temperature. I also collect the water and take it to the lab so I'm able to find out the sediment in the water as well. I use my water samples to vacuum filter 100 milliliters of it and sample it and put it on a pro weight filter, and I dry and weigh it so I'm able to collect the total suspended solids of that subwatershed. I also collect the turbidity with a Hatch turbidity meter to find the cloudiness of each subwatershed. So for my results, this table right here is the mean of the physiochemical of each subwatershed in my Middle White River watershed. It is from October 19th to September 2020. So based on the Ozark Highlands and Delta ecoregions, water quality standards, none of these means actually, none of them go over the standard, which is good. However, if you look at each point value from all my sampling, which is about 52 samples here, the red line is the turbidity standard, you can see that Greenbrier Creek and Lower Salado Creek go over the standard multiple times. However, only Greenbrier Creek is considered impaired from the standards of my ecoregions because more than 25% of that sample exceeds that standard, while the Lower Salado does not go over 25%. So my future works are to keep collecting my water samples till the project ends, which in 2022, update this turbidity with the new year, add graphs of my TSS, and then I analyze total and dissolved nutrients. I collect that in the field and I put them in the freezer, and whenever I'm able to analyze them later down the road, I will be able to get that data. I'm also starting to collect fecal coliforms in my subwatershed, and I will also be collecting my sediment in my subwatersheds to find the toxicity of it. I want to thank my funding sources and also A-State's Ecotoxicology Research Facility, students and staff. Thank you for listening to my project. I hope you have a great day.
Hello, my name is Emily Field, and I am a master's student in the Newman Lee Lab. Snake fungal disease, or SFD, is an emerging infectious disease causing population declines in wild snakes across the eastern U.S. Ophidiomyces ophiodicola is the fungus thought to be the causal agent behind this disease. The first reports of SFD emerged in Illinois in 2006. Since then, snake fungal disease has been documented across most of the eastern U.S. Clinical signs of snake fungal disease include scabs and/or crusty scales with superficial pustules and subcutaneous nodules, cloudiness of the eyes, abnormal shedding, and pinkish red ulcers that are exposed after shedding. Upon histological examination of snakes infected with snake fungal disease, areas of necrosis, granulocytic inflammation and edema, and stain-positive fungal hyphae can be seen. Snake fungal disease has not yet been documented in Arkansas. However, anecdotal reports suggest the disease is present in the state and was potentially present before 2006 when the disease was first reported. Natural history collections are a wealth of data and can be used to detect historical disease prevalence. When used in tandem with contemporary studies, temporal patterns of infectious diseases can be elucidated. The goal of this project is to determine the earliest occurrence of snake fungal disease in Arkansas by performing histological examination of specimens from the ASUMZ herpetological collection. To accomplish this goal, I aim to identify clinical signs consistent with snake fungal disease in preserved specimens, stain and isolate tissues from lesions on these specimens, and examine and identify microscopic signs of snake fungal disease from stained tissues. All snakes in the ASUMZ herpetological collection were fixed in 10% neutral buffered formalin and stored in 70% ethanol. Skin from lesions typical of SFD will be biopsied and decalcified in an EDTA solution. After decalcification, tissues will be dehydrated in a series of graded ethanols ranging from 70 to 100%. Tissues will then be cleared with xylene and embedded into paraffin blocks. Embedded tissues will be cut into serial 4 micrometer sections using a rotary microtome. Sectioned tissues will be stained with a hematoxylin and eosin stain, as well as a Gamori methanamine silver nitrate stain. The H&E stain is used to show general structures and cytology and will allow me to identify areas of necrosis, granulocytic inflammation and edema, and the GMS stain is used to stain and isolate fungal hyphae and will allow me to identify areas where there's infiltration of fungal hyphae with morphologies typical of snake fungal disease. After preparation, slides will be photographed and examined using a Nikon Eclipse E600 light microscope with a Nikon DXM 1200 digital camera. Many scientists now believe we are in the middle of the 6th mass extinction. Snake fungal disease presents a serious threat to snake populations. In a 2006 study, more than a 50% decline in timber rattlesnake populations was documented after the onset of clinical signs consistent with SFD. Additionally, Burbrink, Lorch, and Lips 2017 believe that all 98 snake taxa in the US could be susceptible to this disease. Assessment of the overall health and wellness of wildlife populations is critical to conservation efforts and ultimately can help minimize threats of emerging diseases. This research will confirm or deny the presence of snake fungal disease in Arkansas and will provide us insight into how long this disease has been present in the state. I would like to thank the members of the Newman Lee Lab, Sarah Webb, my committee, Dr. Stan Troth, and my funding. Thank you for coming to my poster talk.
Hi, my name is Brett Hale, and I'm a PhD student in the Molecular Biosciences graduate program at Arkansas State University. Today, I'm going to be presenting part of my dissertation research, which is titled A Systems Approach for the Study of Soybean-Phytophthora soybean Interaction. Soybean is an economically relevant crop worldwide, especially within the United States. In the US, the soybean industry contributes annually greater than $115 billion to the economy, including 350,000 jobs for US citizens. Soybean is the most cultivated crop in Arkansas, and it generates over $1.7 billion for the state's economy on a yearly basis. However, soybean yield is threatened annually by biotic stress, which can cause a yield loss exceeding 400 million bushels in severe instances. Biotic stress refers to living organisms within the growing environment. One prevalent example of a biotic stress that causes yield loss is Phytophthora root and stem rot, which is a disease caused by the oomycete Phytophthora soideae. If you look here in the bottom right, you can see this disease. It causes necrotic lesions near the base of the plant and can cause yield loss up to 40% in severe instances. The oomycetes that cause this disease can overwinter in these tissues and cause an issue for growers for many years. Climate instability exacerbates biotic stress as a whole and really causes an issue with diseases like Phytophthora root and stem rot, because warmer winters mean these pathogens can survive longer periods of time. This creates a need for novel and more rapid crop development technologies. Let's take a closer look at the way soybean interacts with Phytophthora soideae. First, I want to show you an instance of resistance, which you can see here on the left. So if you look at the bottom of this healthy soybean plant, you see these oomycetes, these structures that are colonizing the root system. And if you blow up this bubble here showing an oomycete, you can see these filamental hyphae which extend into the soybean tissue. These hyphae produce proteins which are known as AVR proteins. Soybean can be resistant to this type of attack by producing its own protein known as an RPS protein, which recognizes the pathogen secreted protein in a lock and key mechanism, a ligand receptor system that allows the plant to then elicit a downstream immune response when it recognizes the pathogen protein. However, in instances when soybean is not resistant, either it was never resistant in the first place or the pathogen evolved, you don't get this downstream immune response, this vast transcriptional programming of the plant genes. So in this instance, we're taking a scenario where the pathogen has evolved. And you see we still have the same RPS protein by the soybean plant. However, the proteins have changed shape that the pathogen produces. So because of that, you do not have the lock and key mechanism, and therefore you don't have resistance. So in our lab, we're interested in finding ways for soybean to overcome this lock and key mechanism and be able to fight off Phytophthora soideae by focusing on the downstream immune response, all of the other genes that are important for this that are currently unknown. So in order to do that, we constructed a weighted gene co-expression network. And we did this using previously published RNA-seq data sets, where we were able to look at gene activity when plants were inoculated with Phytophthora soideae and in plants that were healthy. And in so doing, we identified 199 transcription factor hub genes, genes that showed some type of relationship to one another and were most likely involved in some fashion for soybean resistance to Phytophthora soideae. Out of those 199, we were able to prioritize 56, which had been previously documented to be involved in biotic stress response. There were some limitations to our work, however. The study that we used here did not have any biological replicates, so we can't be sure that what we are seeing is reflective of what truly occurs in nature. We only looked at one time point, an optimal time point, but it's essentially taking a snapshot and lacks temporal resolution. So we need to add more time points to really understand this relationship between soybean and its response to Phytophthora soyj. In addition, the model as it stands is currently static. So in order to overcome this, I'm taking multiple approaches to refine our gene co-expression network. First, I'm doing another round of RNA sequencing at the optimal time point, adding biological replicates so that we can truly say if what we're seeing in the lab reflects what would happen in a growing environment in the field of a farmer. The next thing that I'm doing is employing a novel technique known as Capture-Seq. So with Capture-Seq, we're still looking at the activity of genes, but instead of looking at all of the genes in a soybean plant, we're only looking at the ones identified in our network, as well as their potential targets and control genes. So what this does is allows us to work across multiple time points, providing that temporal resolution we spoke of, but doing so in a cost-effective manner that's not computationally intensive. And the way the Capture-Seq experiment will work is first inoculating the plants in the hypocotyl region, which is between the seed leaves here and the root system, to extract RNA from those inoculated tissues. And you can see here, blue and yellow bars, what yellow represent the gene of interest. We then create complementary DNA libraries, which are prepared for sequencing. We then incubate those with probes that are designed specifically for only our genes of interest. You can see a probe here binding to our gene of interest, and then incubating those with beads that have affinity for our probe. So this allows us to only pull out our gene of interest. All the other genes that we don't care about are getting washed away. We can then use this technology to sequence just genes of interest in a high throughput manner. With that, I'd like to thank everyone who has helped me with the research. First, my lab. Second, my committee members. Third, I'd like to thank the Robertson Lab at Iowa State University, who handles all of our plant inoculation, the College of Science and Mathematics faculty and staff, the faculty and staff at the Arkansas Biosciences Institute, the MBS graduate program, and my funding. With that, I thank you for listening to my presentation, and I would be happy to entertain any questions you may have via email or some social media platform. Thank you, and I look forward to further correspondence.
Hi, my name is Veronica Milnes, and I conducted a study on the psychological reaction of Arkansas State University students to the coronavirus. Back in January of 2020, whispers of the coronavirus in other areas of the world reached the university, yet no fear came. It won't end up here, we won't be affected, there's nothing to be afraid of. That's what we all thought. Fast forward to the end of March, beginning of April, when we realized that the coronavirus was a real threat to us. Almost overnight, the university transitioned into online classes, students and faculty were being sent home, social distancing guidelines and mask mandates were being strictly reinforced, and our return to normal was indefinite. So the question is, how did this sudden and mysterious change impact the psychological well-being of students? I assume that the pandemic did indeed have an impact, but of what nature? Were students impacted positively or negatively? And were they impacted a lot or little? These questions circled my head as I listened to the concerns of my friends, family, and peers around me. The easiest way to answer these questions was through Maslow's holistic dynamic theory, which you may know as the hierarchy of needs. Abraham Maslow was a psychologist that evaluated not only our psychological needs, but how our environment interacted with our needs and how our needs interacted with one another as well. Therefore, his theory was the key to understanding our psychological reaction to the coronavirus by evaluating what he proposed as our 5 basic needs. These needs go in a hierarchical order, starting with a physiological needs, such as the need for food at the bottom, the needs for safety, love and belonging, self-esteem, and self-actualization at the top. Based on this hierarchy, I conducted a study to evaluate if and how our satisfaction of psychological needs have changed in response to the coronavirus. Now, what was I expecting to find? Well, ultimately, I thought there would be a negative change in physiological needs and the need for safety. Overall, my survey consisted of general questions about feelings towards health, security, stability, anxiety, and so on. But in creating these questions, I considered that the students being sent home no longer had access to campus cafeteria or the campus store. There was no telling if they were eating properly. Also, businesses and restaurants were closing down indefinitely, so this may have led to students being laid off from their jobs, claiming temporary unemployment, or just generally not having access to the resources they needed. These are the types of things I assumed would influence their answers. Now you might be thinking, what about the other three basic psychological needs? Truthfully, I was expecting to find a positive change in the final three. In this part of my survey, I asked general questions about connection with friends, feelings of confidence, satisfaction with life, and so on. These are the needs I figured would be most influenced by technology, which is what separates our generation from generations past. With technology, students are able to connect with friends, family, classmates, and professors virtually via Zoom and connect with the rest of the world via social media. Technology serves as a connection to others, which would influence the need for love and belonging, and also serves as a creative outlet, which would influence the need for esteem and self actualization. Were my expectations met? Not necessarily. Instead of finding a negative change in some needs and a positive change in other needs, I found a negative change overall for each of the 5 basic psychological needs. Each need was measured on a 5-point Likert scale from negative change to positive change, in which 3 indicated no change at all. Roughly 200 students participated in this study, and as you can see by the graph on the right, they reported the largest negative change in their need for love and belonging and almost no change in their need for self-actualization. This shows that there was indeed a psychological impact as expected, but not of the same nature as I predicted. Instead, students reported a negative psychological impact overall, but not by much. So what does this mean? My interpretation is that technology still makes a positive contribution, but not enough to prevent an overall negative impact from the coronavirus. A virtual connection is not exactly the same as a face-to-face interaction, so technology can only go so far. My guess is without this resource, students would have reported a much more drastic negative change in their need for love and belonging due to the coronavirus. Same goes for the need for safety. Without proper testing and consistent social guidelines to protect each individual, students may have reported a much more drastic negative change in their safety needs as well. At the end of the day, the coronavirus negatively impacted our hierarchy of needs, possibly rearranging it in a way that Maslow would have found confusing. My name is Veronica Milnes, and thank you for listening.
Hello everyone, I'm Sapide Mohammad Hossainpour and I want to present part of my PhD research project, which is about using prenatal Sibnoise as a potential adjuvant for the treatment of triple-negative breast cancer. Why I focus on breast cancer research? As you know, breast cancer is a common cancer among women, and 15% of all breast cancers are classified as triple-negative breast cancer, known as TNBC. Breast cancer is generally classified based on the presence or absence of 3 receptors, and most therapy like hormonal therapy are based on these receptors. But unfortunately, TNBC lacks of all these 3 main receptors, which means it is hard to treat this type of cancer. So it is essential we find the alternative treatment for it. One of the common treatment for TNBC is paclitaxel, which shows kind of toxicity on human body. So we want to check the natural compound which we produce in our lab on TNBC, and also we want to check the combination of paclitaxel with this natural compound. This natural compound we produce in our lab, we call them prenylated Sibnoise. They are produced as a defense mechanism of plants. So we put peanut plants. So we produce the peanut hair root system, and we put the peanut hair root system in a stress situation by adding some chemical. So you can see the media, it turned yellow, and then we extract the prenylated Sibnoise by HPLC. Then we test this prenylated Sibnoise on non-cancer cell line and cancer cell line. So red one shows non-cancer cell line. You can see between all these compounds, Racketing-1 prenylated Sibnoise shows higher toxicity on TNBC cell line and low toxicity on non-cancer cell line. We double check our results by culturing them on cancer cell line and non-cancer cell line. So this is the normal breast cell line. So you can see a lot of cells in our control and same amount in cells when we have our treatments. But in triple negative breast cancer, you can see a lot of cells on control, but you cannot see so many cells when we add treatment to our cells. So you barely can see cells here. The dark blue are showing the dead cells. So we also check the IC50 in different time points for normal cell line and TNBC cell line. You can see that TNBC, when we add arachidine-1, 1 micromolar, they show toxicity in different time points, but in normal cell line, it doesn't show so many toxicity. So based on all these results, we choose the arachidine-1 and start combining it with paclitaxel to see the effect of arachidine-1 on paclitaxel. So you can see the different concentration of arachidine-1 because the paclitaxel IC50 dropped. So we use less amount of arachidine-1, they can drop the IC50 of paclitaxel. Resveratrol also shows dropping in IC50, but we have to use a high amount of resveratrol to be effective. You can see here better, this shows the IC50 of paclitaxel, and arachidine-1 shows how IC50 drop when we use arachidine-1. You can see we need low amount of IC50 in checkboard assay. After that, we check apoptosis too. You can see arachidine-1 shows higher apoptosis in compared to control, but when we combine paclitaxel, paclitaxel and arachidine-1, you see higher apoptosis as well. So based on all these results, arachidine-1 shows higher apoptosis on triple negative breast cancers and higher toxicity, and shows low cytotoxicity on non-cancer cell line and decrease the IC50 of paclitaxel. Based on all these results, arachidine-1 shows effective result on triple negative breast cancer cell line. So thanks for listening to me. Please let me know if you have any questions.
Hello, my name is Brittany Singleton, and I would like to share my poster, Water Quality at 3 Sites Along the Bayou DeView River in Arkansas. The Bayou DeView River in northeastern Arkansas is the main tributary to the Cache River. It is also one of the many tributaries that contributes to the Mississippi River. The water quality of all of the tributaries contribute to the water quality of the Mississippi River. The impairment of a single river can impact downstream water bodies. Large sections of the Bayou DeView are listed as impaired for sulfates, dissolved oxygen, and turbidity. In October 2016, the Arkansas State University Ecotoxicology Research Facility started weekly water quality sampling at 3 sites along the Bayou DeView. Parameters measured were pH, conductivity, dissolved oxygen, turbidity, total suspended solids, total and dissolved nitrogen, and phosphorus. The objective of this study was to determine changes in water quality from upstream to downstream sites along the Bayou DeView River. The null hypothesis was that there would be no statistical variation between the water collected from the Bayou DeView River sites. Under methods, the weekly water samples were collected from October 1st, 2016 to September 30th of 2019 and processed according to the American Public Health Association standards. The statistical analysis for turbidity, total suspended solids, and nutrients were performed in the R programming language. Because the data were not normally distributed, the Kruskal-Wallis test was used. The 3 sites along the Bayou DeView River are each located in a separate Huck 12 watershed. The upstream watershed is the town of Waldenburg, abbreviated TOW. The middle is town of Pittenger, abbreviated TOP, and the downstream is Old Channel Bayou de View, OCBD. There were no statistical differences among sites for the following parameters: total suspended solids, total nitrogen, nitrite, or nitrate. There were statistical differences between the upstream and downstream sites for turbidity, total phosphorus, and orthophosphate. Discussion and action plan. The mean value of 3 years of data can provide overall trends as it covers both base flow and storm events. A variety of trends were observed between the upstream and downstream sites among the 11 water quality parameters measured. For example, water temperature increased in the downstream direction as the riparian areas opened up and the stream order increased, whereas total nitrogen, nitrate, and total phosphorus values decreased from upstream to downstream. This pattern was observed in a previous study in a lower section of the Bayou de View River sampled from 2011 until 2014. This observation coincided with an increase in the percentage of wetlands progressing downstream. Land use within the watersheds surrounding the sampling sites also showed an increase in woody wetlands, as seen in Table 1. Although both turbidity and TSS increased from the upstream site to the middle site, the mean value at the downstream site was smaller than the middle site. Since this downstream river site, OCBD, has 23% woody wetland, this decrease in turbidity and total suspended solids might be due to the filtering effect of forested wetland areas, as indicated by previous studies. Since phosphorus can bind to soil particles, a reduction in sediment may correlate with a decrease in phosphorus. High levels of nitrogen and phosphorus in the Mississippi River are contributing to the Gulf of Mexico hypoxic zone. Reductions in the amount of nutrients flowing downstream in the Bayou de View River can help reduce the nutrient load heading to the Gulf of Mexico. Future work on this project will include continuing collection of weekly water samples until September 30th of 2022 and analyzing data for short and long-term trends.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello, my name is Landon Bonner, and today I'll be presenting what elements of caregiving influence teen drug use. My fellow authors are Emily Burke and Emily Weaver, and our mentors were Dr. David Sarnio and Dr. Sharon Davis. So first, some background. Millions of Americans use drugs and alcohol. Why? Well, drug use can commonly start in adolescence. And what's the reasoning behind that? Well, behavior displayed by the primary caregiver, the one who's most responsible for care and responsibility of the caregivee, can play a key role in the development and play a key role in discouraging specifically teen drug use. So the purpose of our study was to see exactly which factors of behavior displayed by the primary caregiver during upbringing correlated with teen drug use. And our hypothesis was that there would be a relationship between teen drug use and the different factors of upbringing. So we know that the behavior by the primary caregiver has a role, but we want to see exactly which factors and which behaviors specifically have the best and most obvious role. And so what we had was a questionnaire of 111 psychology student participants that looked over caregiver and drug use questions, 2 different sections. And so the first section, the caregiver section, had 32 questions, which looked over 3 different factors. The first factor, caregiver support, looked at how involved and supportive the primary caregiver was. And so, for example, my primary caregiver provided emotional support. My primary caregiver was involved in education. And so the next factor, caregiver respect, looked at how responsive the caregiver was. My caregiver listened to my point of view. My caregiver allowed me to make my own decisions. And so the next factor was caregiver strictness, and that looked at how restricting the primary caregiver was. So for example, they emphasized tradition, they expected strict obedience, stuff like that. And so the next section looked at drug use. And so it contained 10 drug use questions in total, and it looked over 2 different factors itself. The first factor was common drug use. And common drug use included drugs such as marijuana, alcohol, tobacco. Hard drugs included opioids, hallucinogens, and other stuff. And so what you see in our results was that caregiver respect was significantly correlated with both hard and common drugs. And with caregiver strictness, neither was related to hard. Caregiver strictness was not related to either hard or common drugs. And with caregiver support, you see that both scores are significant with both hard and common, and was even stronger than it was for respect. And so what you see in the regressions, and this first one is common drugs, is that the beta for caregiver support was much stronger than it was for either respect or strictness. And you see the exact same thing with hard drugs. Caregiver support stood out from the other 2 factors. And so in conclusion, caregiver support and respect correlate with both hard and common drug use. However, caregiver strictness did not. And caregiver support was decisively the most important correlate in this study. And so what that means is that the more the primary caregiver was involved in the lives of the caregivee, the more they provided emotional support, the more they were there for them, the less drugs that they used. And the more respect and how responsive they were also had an effect as well. However, emphasizing tradition, for example, the strict obedience, the caregiver strictness did not necessarily have a key role. And so what all this means is that behavior displayed by the primary caregiver during upbringing does indeed matter. It does have a relationship with teen drug use, and they do have a role. And so what you see is that behavior by the primary caregiver is important, and it does have an impact. This is some of the literature used and past literature on the topic, and thank you.
Hello, my name is Sarah Brown, and I've been doing research on the impact of educational television under the supervision of Dr. David Sarnio. Anyone with kids or anyone who watched TV as a child is likely familiar with some kind of educational show. There's so many out there to choose from, and many have been around for a really long time. Producers of these shows have long thought that television has the capacity to be impactful in the lives of their child viewers. Shows like Sesame Street and Mr. Rogers' Neighborhood have been around since the 60s, and programs serving similar purposes, namely to provide educational benefits to children and prepare them for formal schooling, still exist today. Sesame Street is even one of them. They've been producing new episodes and content since the late 60s. Studies conducted on the short-term effects of these educational programs have found that they can increase literacy skills, perspective taking, emotional regulation, and they can decrease the amount of negative stereotypes children may hold against other groups. One study that looked at census data from 1969 even found that children who lived in areas where Sesame Street had increased coverage were more likely to be in a grade fitting for their age. Far fewer studies have been conducted on long-term effects, but the study by Kearney and Levine that used old census data found that children who had been living in areas where the signal for Sesame Street was especially strong when they were viewers of the program were 14% more likely to be in a grade fitting for their age in middle and high school as well. So, what did we do? Because research on long-term effects of educational television is so limited, we decided to look at college students' perceptions of the long-term impact of the educational programs they watched in childhood. We looked at their perceptions on 4 educational programs: Blue's Clues, Dora the Explorer, The Magic School Bus, and Sesame Street. We chose these 4 programs based on the responses participants gave to a survey question asking them which program they viewed most often in childhood. That was part of a pilot study. We wanted to know how they believed these programs impacted them on cognitive, social, and emotional dimensions. Participants chose which of the 4 programs they viewed most often in childhood, and they answered the extent to which they believed that particular program taught them some such skill on a scale of 1 to 5, with 1 being strongly disagree and 5 being strongly agree. These are the skills we asked participants about, organized by dimension. Some of the skills we asked about for the cognitive dimension were how to read, how to count, and how to problem solve. Some of the skills we asked about for the social dimension were how to share with others, how to solve problems without violence, and how to work cooperatively with others. And some of the skills we asked about for the emotional dimension were how to recognize your emotions, how to control your emotions, and how to recognize how your emotions affect others. We asked about cognitive, social, and emotional skills because educational programs have been found to impact each of these skills in the short term, and we want to see if that may hold true for the long term. So, what did we find? Overall, college students remember what they watched, and they generally believe the programs they watched had a positive impact on all 3 dimensions. The highest perceived impact was on social skills, then cognitive, then emotional. This did vary by program, and Sesame Street rated the highest on all 3 skills. The Magic School Bus rated the lowest on cognitive skills, and Blue's Clues rated lowest on social and emotional skills. So, why does any of this matter? Well, this implies that these programs do have some kind of long-term impact, particularly on social skills. It seems that children's educational television may be more impactful in the long term than we anticipated. College students are remembering what they viewed, and they're remembering it as being impactful to them on all 3 dimensions, but again, particularly on social skills. Additionally, results from this study can be used by television developers, producers, and writers to build on the past success of these programs we studied and create more impactful content and programs for future child viewers. Here is some contact information, and here are our references. Thank you.
Hello, I'm Emily Burke, and I'm presenting Adolescent Drug Use and Parental Social Media Monitoring. My fellow authors are Landon Bonner, Emily Weaver, Dr. David Sarnio, and Dr. Sharon Davis. So first, I want to talk about why this is important. So many adolescents are putting themselves at risk by using drugs. And just to give you an indication of how many adolescents are doing this, according to Monitoring the Future in 2018, 55.3% of high school seniors had used alcohol in the last year, and 35.2% of them had used marijuana in the last year. That is an alarming rate of adolescents using drugs before they've even graduated high school. Something that's very prevalent in the lives of many adolescents nowadays is social media. And based on past research, social media has actually been found to be able to influence the decisions of adolescents. And this is just because when an adolescent sees a behavior being done on social media, then they may be more likely to facilitate that behavior themselves. And that gets us into our purpose. So social media theoretically has to filter through the parents. So that's why our research question was, is there a relation between parental social media involvement and adolescent drug use? Well, the adolescent's decision to use drugs. And we did expect to find a relationship. So we put out a questionnaire for the research experience component that is a part of entry-level psychology classes, and we ended up getting 111 participants. The participants were asked questions about their social media drug exposure, which is just, were my friends using drugs on social media, and was I seeing that? And they were also asked questions about their parents' involvement in their social media. And involvement was operationalized as monitoring and awareness. And so monitoring would be, did my caregiver block certain sites? And awareness would be, was my caregiver aware of my image on social media? And then the participants were asked questions about their drug use. They were asked about their frequency of use of alcohol, marijuana, and tobacco from the ages of 15 and 16. So we found that social media drug exposure and adolescent drug use were significantly correlated, as you can see. We also found that caregiver monitoring was correlated to adolescent drug use. It was more weakly correlated than social media drug exposure, but we did find a small correlation there, a negative correlation. And caregiver awareness was not found to be correlated with adolescent drug use. So in conclusion, what we learned from this is that social media drug exposure was most significantly related to adolescent drug use, and that does replicate other research. And although caregiver monitoring was not as strongly related, we did find a relationship there, and we found no relationship between caregiver awareness and adolescent drug use. However, this is still really important because what we found is that what adolescents have access to on social media is important and it does matter. And parents and caregivers have the opportunity to pay greater attention to what their adolescents are looking at on social media, and those efforts may curb adolescent drug use and may prevent it. These are the references that were used for this presentation. Thank you very much.
Hi, my name is Traylee Ellington, and this is my presentation on teaching writing processes in middle school. I started with the question, what inspires people to write, which turned into what steps can teachers take to encourage and improve writing, and what exercises and methods in writing do students benefit the most from. And I was led to 3 different methods of teaching writing. 1, using images to inspire writers, 2, using models from teachers and peers as examples, and 3, using mentor texts from various sources. Firstly, on using images to inspire writers, I want to touch on Linda Rife's book, Seeking Diversity, more specifically her chapter covering the art of literature, where she uses a workshop that connects art and literature in a very creative way. In this workshop, she had her students pick a book they felt had a big emotional impact on them and turn it into a piece of art, which ended up resulting in a much deeper analysis of the story and its meaning than the students originally had. Now, Crystal Allen is an author. She writes books that focus on the middle school demographic, and she goes to schools and does writing workshops with middle school students. And her workshop is essentially the opposite of that. After describing what a story is and what pieces a story needs to be a story, she puts a image on the board and says, okay, you have 45 seconds to write a story based on this image. And these students do. They write brilliant stories. You can go on her website, crystalallenbooks.com, and go to her Strike Writers contest, where students submit their final draft of these stories for the competition, and 13 from each school she goes to are chosen as winners. And all of their stories are listed on there. And they're very brilliant stories because this picture, these images, art, gives students a scene to work with, a scene without context that they can build on and expand on. And it provides fuel for thought. Secondly, using examples from teachers and peers, Nancy Atwell, in her book, In the Middle, Understanding About Writing, Reading, and Learning, discusses how students tend to write better, write more, and have more enjoyment in it when they write about topics they enjoy and have a lot of knowledge on. And I believe this is because students are confident in writing about these topics. It's something they're familiar with, it's something they enjoy, and so they've learned a lot about. And confidence equals motivation. And when a student has motivation to write, they have motivation to improve. Building confidence is a very important part of teaching writing. I interviewed a 6th grade writing teacher, Miss Lindsay Hart-Smith. She also believes that confidence is a big part of being able to write and encouraging students to write. In her classes, she often encourages students to share their own works with their peers and classmates in order to get positive feedback from them and to boost their confidence in A, speaking in front of a classroom, which is intimidating for many students, and B, in talking about their writing. And also, whenever they're doing lessons, she gives examples of her own work and works and does the same exercises as they do. So that way they have a teacher's example to tell them exactly what is needed, what they're expected to do, and to help guide them in the lesson. Crystal Allen, in her workshop, she asks the teachers to stand behind students who typically struggle in class when they raise their hand to share their works. So that way she may call on them and prioritize calling on them to share their works. So that way they also may receive that positive feedback from peers in order to boost their confidence and hopefully encourage them to continue writing. So lastly, using mentor texts as a guide for writing. Mentor texts are basically any text, books, poems, lyrics that a student can take pieces from in order to guide their own writing. In Miss Lindsay Hartsmith's lesson on personal narratives, one of the mentor texts she uses is called The Iguana in the Bathtub by Anne Doughton. And after analyzing the paragraph, she has students write their own personal narrative with the same structure. So they start a sentence that goes, when this happened, that happened. And then they have two sentences that follow it to further describe the scene. And this way of using mentor text gives students a solid structure for their writing while allowing them the creative ability to fill it with whatever content they want. It is also very important to use diverse mentor texts. In the book list webinar I attended, author and educator roundtable, culturally responsive books in the classroom, the panelists, Candice Ilo, Jessica Kim, Tori Maldonado, and Simranjeet Singh, all stressed how important it was for them to be able, as children, to be able to see themselves in what they were reading and what they were seeing. There is also a TED Talk, The Danger of a Single Story, which discusses at length how we write what we read, which makes it very important to choose diverse mentor texts and diverse examples in popular literature because your students, in order to connect with their writing and to be encouraged to write, need to be able to see other people like them, not only in literature, but who have also succeeded in writing. So that wraps up my presentation. I hope it was informative, and I hope you all have a very nice day.
Hello everyone, my name is Megan Gunnels, and today I will be doing a brief presentation over my honors thesis project. So in March of 2020, the COVID-19 pandemic closed down Arkansas State University as well as many other schools across the country, and we began pursuing remote learning opportunities. Social distancing was happening, quarantine was happening, and I began to notice through this that my coping styles as well as others in my family and that I was around had changed from previous types of coping, and so I wanted to do my honors thesis project over this and see if the COVID-19 pandemic had impacted students' coping styles and see if personality played a role in that. A lot of the literature for coping had discussed coping after natural disasters and wars, and I wanted to adapt some of that to involve coping after a pandemic. So I wanted to see if there's any type of correlation between coping and different aspects of personality, and included a locus of control measure as well. So I used 3 already established measures: the Carver Brief Cope Scale, the Roeder's Locus of Control Scale, and the 10-item personality inventory. And so the survey was given through the A-State SONA system, and we had 144 participants who all received the same survey, a consent page, a 28-item brief COPE scale, a 10-item personality scale, an 11-item locus of control scale, and demographic questions. And so we had over 130 significant correlations between all of these measures, but for the sake of time, I will only go over a few. There was a positive correlation between denial and behavioral disengagement, which can be assumed because the more a person is disengaging from society and their behavior is disengaging, you can see that they are partaking in more of a denial coping mechanism, just denying the whole pandemic, denying what they're doing, it's not really happening, and just disengaging from that. There's a positive correlation between emotional support and venting, which is interesting because emotional support was something that was highly needed during the social distancing and the quarantining. So if a participant felt that they had a strong emotional support system, then you're seeing them partake more in that venting coping style, which is really interesting. There is a positive correlation between neuroticism and locus of control. So the personality style of neuroticism is positively correlated with an external locus of control. So the more of an external focus that a participant is taking, the more you can see that neuroticism personality style. And there's also positive correlation between substance use and locus of control, which is interesting to note because substance use can quickly turn into substance abuse. And so if we're seeing more of these students with a external locus of control partaking in substance use, that can quickly turn into abuse if not careful. And so it's important to note that positive correlation. These are our participants. And so, as I said, there's 144 participants. And one of the things that I should note is that majority of our participants were Caucasian female. And so I would love to see the survey done with more minority students and non-traditional students. Another thing is that Arkansas State University did go back to campus in August, so for the fall 2020 semester. And so I would love to see the survey in universities where they continued remote learning through the fall semester and into the spring semester and see if coping was different for those types of students at that university. As far as future research, I think that the effects of these measures can be really important for the future ramifications of higher education. I think that changes in students choosing remote learning options could be seen as far as their coping. So students who really enjoyed remote learning, had positive coping mechanism styles, would probably prefer remote learning versus students who had very negative coping and would prefer the face-to-face instruction. There also could be changes in students who are even choosing to pursue higher learning opportunities in general. High school students may have chosen to take a gap year between high school and beginning their undergraduate degree, as well as students who were in their undergraduate degree during the pandemic who may have chosen to not pursue a graduate program, or students who chose to take a gap year. So I think that could be interesting to look at based off students' coping mechanisms. So I just want to thank you all for your attention today. And I'd also like to thank my mentor, Dr. Jessica Curtis, for assisting me throughout my honors thesis project for this past year. So once again, thank you all for listening.
Hi, my name is Cameron O'Connor. I am an undergraduate psychology student, and I'm going to be talking about race conversations in the classroom. So over the past year and a half, we've experienced a global pandemic. We've seen a controversial election in its aftermath, but we've also seen a series of high-profile events of violence towards Black Americans, such as the killing of George Floyd and the shooting of Breonna Taylor. So emotions are running high. We've seen Black Lives Matter protest and social media campaign in response to these instances. And now more than ever, race is becoming a topic of conversation. But Black and white Americans view the current climate differently. Black Americans feel like there's still much work to be done to address racial injustices, while attitudes of white Americans remain largely unchanged since 2019, or since before we started seeing a lot of these events happen. So we need to have more discussions about race. We need to have these conversations to promote understanding across racial lines. And a transition to the classroom could be ideal to do that and to shift attitudes for the better. But how would our students feel about these conversations? So Daryl Tsu suggested there are 3 main protocols or characteristics regarding race talk. Typically, race can evoke negative and strong feelings, and the academic protocol suggests that students are encouraged to suppress those feelings if they're intense or strong for the sake of the academic environment or the classroom. The politeness protocol suggests that students will prioritize respect and being polite, and to do that, they typically avoid uncomfortable conversations and topics, and race is usually one of those topics. And last, the colorblind protocol says that students overlook racial differences. So we wanted to see if there were differences in how white and Black students perceive these protocols in the classroom. The survey was 31 items and presented to lower level psychology students. And to supplement the African American population, it was presented to historically African American fraternities and sororities. The items were on a 5-point Likert type scale from strongly disagree to strongly agree. And below we have some of the items and then the protocols that they are representing. So for the politeness protocol, we have students should be respectful of each other during conversations about race. And my feelings about race are too intense to be shared. The academic protocol, the classroom is not a place to share my opinions about race, and conversations about race should be discouraged in the classroom. And the colorblind protocol is during conversations about race, I am colorblind and I do not see my peers in terms of race. Only the self-identified African American and white students were analyzed for this survey. So starting with the politeness protocol, we see that both groups of students strongly agree. And again, that politeness protocol is where students are going to prioritize being respectful and polite regardless of their beliefs. So we see that both of our students understand the importance of being respectful regardless of what the topic of the conversation is. When we get to the academic protocol, though, we start to see a little bit of difference. The academic protocol is where students feel that they should suppress their intense feelings for the sake of the academic environment or the classroom. Our African American students strongly disagree with this protocol, suggesting that they don't believe that they should suppress their intense feelings, and that they do feel like the classroom is the place to share those feelings, while our white students fall right around that mean, not knowing whether to agree or disagree with the academic protocol. And lastly, the colorblind protocol is where we see our biggest difference. And that colorblind protocol is where students are asked whether or not they see their peers in terms of race or whether or not they are colorblind. Our African American students strongly disagree, meaning that they do see their peers in terms of race. And our white students strongly agree, saying that they don't see their peers in terms of race or that they are colorblind. So why does this matter and why is it important? So our survey gave us a little bit more insight about how our students feel, but our educators and teachers are just as important when it comes to these conversations. These conversations are becoming unavoidable and they're happening more and more often. So our teachers need to be open and willing to address these problems head on and not treat race as the elephant in the room. And we also want our classroom environment to feel safe for our students. We want our students to feel free to talk about the negative aspects that are typically associated with race, such as injustices and inequities, but we want the classroom to be safe and open to talk about and highlight the positive aspects of race and diversity as well.
Hi, my name is Mary Reed. I'm a student here at Arkansas State University in the psychology department. Emily Burke and Landon Bonner are also co-authors on this research project, along with our mentor, Dr. Sarnio and Dr. Riondolio. We are conducting a research study to see if dogs with black coats are truly less preferred than other dogs. When a dog is brought to the animal shelter, it often has 3 fates. 1, it could be adopted. 2, it could be reclaimed by its original family. Or 3, it will be euthanized. Black dogs are commonly left in animal shelters for longer periods of time than other dogs, and longer stays in animal shelters often leads to euthanization. We're going to collect survey data to help determine if the black dogs are truly less preferred because of their coat color, or could it be some other reason? Each of our participants will be completing a survey. They will be shown multiple different breeds of dogs, but each breed they'll see 3 different colors. They'll be asked to rate these dogs on a 5-point rating scale, strongly agree to strongly disagree, for 4 questions. The 4 questions will be examining adoptability, likability, features, and color. So, for example, if the participant were rating labs, they would get a picture of a chocolate lab, be asked 4 questions, and then they would be shown a different breed of dog, asked the 4 questions, and then we go back to the lab in a different color than before and ask the 4 questions again, therefore to see if it's the breed or is it truly the color that is less preferred. So which dog would you consider adopting? A, B, or C? All of these dogs are the same breed, just different colors, and we believe that there truly will be a preference based on color. So even this question, which color do you prefer? Black, tan, or white? Again, it's the same breed, we're just mixing the colors up. So what were your answers? If you chose anything other than black, that's actually the responses that we are expecting to get. We think that the responses of our survey will reflect the fact that dogs with black coats are truly less preferred than dogs with other coats. We're still in the process of collecting this data, so we're looking forward to seeing if it really is because of their coat color. This will help us increase adoptability rates in shelter dogs and also decrease euthanization rates. If we can understand why black dogs are being left in animal shelters for longer periods of time than other dogs, hopefully we can increase adoptability rates or decrease euthanization rates. Again, my name is Mary Reed, and I thank you for your time.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hi, I'm Elise Nooner.
My name is Tinsley Clark.
And I'm Hannah Tisdale, and our project is called Using Scientific Principles to Address Barking in Shelter Dogs. Establishing the social significance of behaviors that might be targeted to change is an important aspect of the applied research process. There are ethical considerations one must take when designing an intervention that will change the behavior of another organism. There is an accountability responsibility that comes with the power of adjusting behavior. Ethical standards require the formal evaluation of potential target behaviors prior to introducing an intervention that will change behavior. You must answer the question, will the change be beneficial to the organism? Is the target behavior important to the organism and relevant to their well-being? We selected barking behavior in shelter dogs as our target behavior. The social significance of this project is how shelter dogs will be impacted by our research. Our goal is to reduce barking behaviors of dogs in humane societies to increase their chance of being adopted. Our research team has come to the conclusion that reducing this behavior in shelter dogs could improve their chances of adoption and therefore their quality of life. This is also extremely important because the more dogs that get adopted, the more room the Humane Society has to help other dogs. Also, this research will impact the volunteers who work with the animals by allowing them to see the interventions we use to reduce the behavior and be able to implement those techniques long after our research team is gone.
So, barking as a behavior is operationalized by a set of criteria. This set of criteria is set in place in order to accurately determine the presence of the behavior of barking. This operational definition also ensures that our measurement of behavior is consistent between observers. Canine barking operationalization includes the following characteristics: open mouth vocalization, abrupt in onset in contrast with a whine, short in duration in contrast with a howl or bay, may occur in rapid repetition, and relatively loud in contrast with a whine. The following characteristics are excluded from our definition: closed mouth vocalizations, open mouth with no vocalization, such as seen in pants, and gradual onset, such as seen in a how or they.
One of the important decisions we had to make before we could begin measuring our target behavior is the type of measurement strategy that we will use. In order to accurately record our observations of dogs barking, we had to decide whether a partial or momentary interval recording strategy was best. We chose partial interval recording. In partial interval recording, the observation period is broken up into 10-second intervals. If a dog barks during the current 10-second interval, a single tally mark is made indicating the target behavior occurred. A dog only has to bark once in an interval for the behavior to be counted as occurring during an interval. In contrast, momentary interval recording also involves breaking an observation period down into 10-second signaled intervals. The target behavior is recorded as occurring only if it is taking place when the 10-second signal sounds at the end of the interval. For instance, if the dog barked every 10 seconds on the dot, a tally mark would be recorded. One pro of partial interval recording is that it provided us an estimate of repetition and duration of each dog's barking behavior. One con of using partial interval recording was that it required our full undivided attention. It was quite hard for us to keep up with each dog's bark just by watching the video. Therefore, we each downloaded an app called Insight to help us record and tally each bark that the dog was making. This made partial interval recording much easier.
So overall, the takeaway from this project is measurement matters.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello, my name is Lindsay Bowling, and for my research study, I'm going to evaluate the effects of a carbohydrate mouth rinse on the performance of soccer-specific skills. Carbohydrate mouth rinse has been shown to enhance aerobic activity lasting longer than 30 minutes, as well as enhance some anaerobic and skill-related tasks. However, the effects of a carbohydrate mouth rinse on soccer-specific skills has yet to be evaluated. So therefore, the purpose of this study is to evaluate the effects of the carbohydrate mouth rinse following an early morning soccer practice on the execution and accuracy of soccer-specific skills such as passing, agility, juggling, and shooting in fasted collegiate female soccer players. For this study, the Arkansas State NCAA Division I female soccer team has agreed to participate. We will have a total of 3 sessions, each separated by 7 days. The first session will be our control session, during which we will collect baseline data for the soccer-specific skills. And then the 2 experimental sessions will be counterbalanced in design. We will ask participants to keep a dietary record 24 hours prior to that first experimental session and to replicate that 24 hours before that second experimental session. They will also be provided with a standardized meal the night before each experimental session and then be asked to fast overnight. So the experimental sessions will be double blind and counterbalanced in design. Participants will arrive after that overnight fast in the morning, and we will collect a finger capillary blood sample to assess their blood glucose levels. They will then participate in a 5-minute coach-led warm-up followed by a 1-minute rest period. They will then perform a 30-minute high-intensity coach-led soccer practice followed by another minute of rest. And during this rest period, they will rinse their mouths for 10 to 15 seconds with either the carbohydrate solution or the placebo solution, and then expectorate it back into a cup to be reweighed to make sure that none was ingested. And they will also identify their thirst sensation and rating of perceived exertion during this rest period. And then following that rinsing procedure, they will perform those soccer-specific skills that we are assessing during this study. We will also be monitoring their heart rate to determine the intensity at which they're working during the experimental sessions. The passing drill will consist of participants standing 8 to 10 meters away from 2 cones separated by about half to 1 meter's distance. Participants will kick a stationary ball 5 times with their right foot and 5 times with their left through the 2 cones, and the ball must not make any contact with the cones for the pass to be counted as successful. The agility test will be a standard t-test. Participants will perform 2 trials of this, and the best of 2 trials will be used for data analysis. We'll be using a photocell timing system to time the t-test. For the t-test to be counted as successful, participants must touch the base of the cones as they arrive to the specific point with their hand. They must also face forward during the entire test, and during the shuffles from points C, B, and D, participants' feet must remain parallel. They cannot cross over one another. The juggling test will be a 1-minute test during which participants will use their feet, thighs, chest, head, or a combination of all to juggle a soccer ball. And the number of times that the ball makes contact with their body will be counted during that minute and used for data analysis. The shooting drill will replicate a standard soccer penalty kick. So for this, participants will stand 11 meters away from a standard soccer goal. We will place cones 1/2 to 1.5 meters in on both sides of the goal. Participants will use their dominant foot to kick 5 shots to the left side of the goal and 5 to the right side of the goal. For the shot to be counted as successful, the ball must remain in the air as it reaches the goal. It cannot roll on the ground, and it must enter that designated space between the side of the goal and the cone. So this study is ongoing. We will collect data in April, but we do have some expected results. We expect that the carbohydrate mouth rinse will improve soccer skill performance when compared to the placebo trial. We also expect that rating of perceived exertion, thirst sensation, heart rate, and glucose levels will be similar across interventions. And if performance is improved, coaches should consider implementing a carbohydrate mouth rinse during high-intensity games and practices when fatigue is likely to occur. And it could also be used as an alternative to consuming a pre-game meal or beverage to help avoid that gastrointestinal discomfort that sometimes occurs during high-intensity activities. Here are some references. Thank you for listening. I hope you have a great rest of your day.
Hi, my name is Veronica Milnes, and I conducted a study on the psychological reaction of Arkansas State University students to the coronavirus. Back in January of 2020, whispers of the coronavirus in other areas of the world reached the university, yet no fear came. It won't end up here, we won't be affected, there's nothing to be afraid of. That's what we all thought. Fast forward to the end of March, beginning of April, when we realized that the coronavirus was a real threat to us. Almost overnight, the university transitioned into online classes, students and faculty were being sent home, social distancing guidelines and mask mandates were being strictly reinforced, and our return to normal was indefinite. So the question is, how did this sudden and mysterious change impact the psychological well-being of students? I assume that the pandemic did indeed have an impact, but of what nature? Were students impacted positively or negatively? And were they impacted a lot or little? These questions circled my head as I listened to the concerns of my friends, family, and peers around me. The easiest way to answer these questions was through Maslow's holistic dynamic theory, which you may know as the hierarchy of needs. Abraham Maslow was a psychologist that evaluated not only our psychological needs, but how our environment interacted with our needs and how our needs interacted with one another as well. Therefore, his theory was the key to understanding our psychological reaction to the coronavirus by evaluating what he proposed as our 5 basic needs. These needs go in a hierarchical order, starting with a physiological needs, such as the need for food at the bottom, the needs for safety, love and belonging, self-esteem, and self-actualization at the top. Based on this hierarchy, I conducted a study to evaluate if and how our satisfaction of psychological needs have changed in response to the coronavirus. Now, what was I expecting to find? Well, ultimately, I thought there would be a negative change in physiological needs and the need for safety. Overall, my survey consisted of general questions about feelings towards health, security, stability, anxiety, and so on. But in creating these questions, I considered that the students being sent home no longer had access to campus cafeteria or the campus store. There was no telling if they were eating properly. Also, businesses and restaurants were closing down indefinitely, so this may have led to students being laid off from their jobs, claiming temporary unemployment, or just generally not having access to the resources they needed. These are the types of things I assumed would influence their answers. Now you might be thinking, what about the other 3 basic psychological needs? Truthfully, I was expecting to find a positive change in the final 3. In this part of my survey, I asked general questions about connection with friends, feelings of confidence, satisfaction with life, and so on. These are the needs I figured would be most influenced by technology, which is what separates our generation from generations past. With technology, students are able to connect with friends, family, classmates, and professors virtually via Zoom and connect with the rest of the world via social media. Technology serves as a connection to others, which would influence the need for love and belonging, and also serves as a creative outlet, which would influence the need for esteem and self actualization. Were my expectations met? Not necessarily. Instead of finding a negative change in some needs and a positive change in other needs, I found a negative change overall for each of the five basic psychological needs. Each need was measured on a 5-point Likert scale from negative change to positive change, in which 3 indicated no change at all. Roughly 200 students participated in this study, and as you can see by the graph on the right, they reported the largest negative change in their need for love and belonging and almost no change in their need for self actualization. This shows that there was indeed a psychological impact as expected, but not of the same nature as I predicted. Instead, students reported a negative psychological impact overall, but not by much. So, what does this mean? My interpretation is that technology still makes a positive contribution, but not enough to prevent an overall negative impact from the coronavirus. A virtual connection is not exactly the same as a face to face interaction, so technology can only go so far. My guess is without this resource, students would have reported a much more drastic negative change in their need for love and belonging due to the coronavirus. Same goes for the need for safety. Without proper testing and consistent social guidelines to protect each individual, students may have reported a much more drastic negative change in their safety needs as well. At the end of the day, the coronavirus negatively impacted our hierarchy of needs, possibly rearranging it in a way that Maslow would have found confusing. My name is Veronica Milnes, and thank you for listening.
Greetings from the College of Liberal Arts and Communication at Arkansas State University. My name is Carl Cates. I'm Dean of the college. Today, I want to welcome you to our Creative State presentations. This college is the home to fine arts, media and communication, humanities, and social sciences. Our students are active in our outreach to the community and in presentation of their research. Creative State is one of those venues. We hope you enjoy what you see and hear today.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello, my name is Rachel Blocker, and I will be talking about the effects of divorce on children's communication through attachment theory. In the United States, a couple is divorced nearly every 13 seconds, resulting in a nationwide divorce rate of almost 50%. This in turn means that a large number of children in America are children of divorce. When a child experiences a divorce in their family, they face a multitude of disadvantages, including worse performance in school, they're less likely to attend a secondary school, they're more likely to develop depression and substance abuse issues, they're more likely to have issues with authority, and they struggle to build and maintain meaningful relationships. So attachment theory was developed in 1958 by Bowlby and Ainsworth, and they deduced that you could understand a child's attachment style through their relationship with their caregiver. They developed 3 different kinds of attachment styles. They are secure, avoidant, and anxious ambivalent. A secure child often receives the correct amount of care, resulting in a positive view of themselves and the world. Avoidant children, they receive too much or too little care, resulting in a negative view of the world. And anxious ambivalent children receive inconsistent levels of care. So one day they may have all of the attention from their parents, and the next day they may have none. Children of divorce are likely to fall in either the avoidant attachment style or the anxious ambivalent attachment style. Anxious ambivalent children are also likely to develop negative views of the world. However, they still may develop positive views of the world. It depends on the severity of the inconsistency in their care. So as these children grow up, these attachment styles follow them. In 1990, Bartholomew looked further into attachment styles in adulthood, and she defined 4 categories stemming from Ainsworth's and Bowlby's initial 3. They are secure, preoccupied, dismissive, and fearful. Adults who have a secure attachment style have a positive model of themselves and others, and they are more likely to maintain healthy relationships as they have better communication and conflict management skills. Preoccupied individuals, they have a negative model of themselves, but a positive model of the world. This means that they are very reliant on affirmation and they cling to relationships. They face issues communicating, and they tend to focus on themselves in arguments, and they're often overly controlling and emotional, and that stems from anxious ambivalent attachment style in childhood. The fearful style of attachment in adulthood means that the adult has a negative view of themselves and others. They fear being let down, often to traumatic events in the past, like their parents' divorce. They restrain themselves from disclosure, and they have less developed social skills because they avoid people. And that stems from anxious, ambivalent, and avoidant attachment styles in childhood. And then finally, dismissive. Adults, they see themselves positively, but others negatively. This results in them being self-sufficient and avoiding relationships. They also tend to value material items over relationships, and that stems from avoidance. So in conclusion, children who are affected by divorce in America have longstanding psychological issues that follow them into adulthood, which ultimately impacts their chances for happiness in adulthood. These children are at a large disadvantage compared to children whose parents stay together. Therefore, it is the role of parents, teachers, peers, counselors to understand this, to better understand how to help children of divorce to develop healthy attachment styles so that they may find happiness in adulthood, and also to build better relationships in adulthood. Attachment theory is useful to this study as it helps you understand why children of divorce face more issues and the psychological processes and pressures that they face following divorce. These issues that children of divorce are faced with are often carried into adulthood and can ultimately affect their happiness, as I previously said. And it emphasizes the need for extra support for children of divorce to help create healthy coping mechanisms and ultimately healthy attachment styles so that they have the best chance that they can have in adulthood for happiness. Thank you.
I wish I could just skip the part where I don't know the right thing to do and get to the part where I do. This quote illustrates the internal conflict many women experience when confronted with social standards and expectations around the world. First, I'm going to be talking about my text for my analysis. Secondly, I will discuss my theory and my methodology for my analysis. Thirdly, I will be discussing my analysis as a whole. And finally, I will be providing concluding remarks regarding my research analysis. My artifact is from the Showtime series Shameless and will be focusing on the character Debbie Gallagher. Debbie is a teenage working mother who lives on the south side of Chicago and is trying to be her own woman and set a good example for her daughter while having to conform to social expectations. This artifact shines light on the ever-changing social expectations placed on women and reveals different gendered factors that most strongly impact women and debunks their credibility so that change can be brought forth in society. For my methodology, I have used textual analysis. A woman's social role and personal perspective in life are harsh realities that women experience that can be debunked and altered by disciplining gender, breaking gender norms, and rejecting heteronormativity. For this presentation, I will be focusing specifically on breaking gender norms, specifically for women. Gender norms are the societal expectations that are placed on both men and women to perform a certain way, dress a certain way, and many other strict standards. In Shameless, Debbie is known for breaking these gender norms. For example, she's a mother that was constantly pressured by her peers as a teenager to do what is best for her child and not work long hours so that she could spend time with her daughter. But instead, she breaks this expectation and goes to school in Southside Chicago to become a welder in downtown Chicago so that she can provide for her child and give her a good life. She also breaks gender norms by playing the role of both mother and father. Whenever Debbie found out she was pregnant, she was still in high school and was already in a rough and kind of teetering relationship with her boyfriend, Derek. When she told him that she was pregnant, he broke up with her and told her that he just wasn't ready to be a father and his future was way too important that this could not get in his way and stand in his way. Derek been enlisted in the military and didn't want anything to do with Debbie's daughter, Franny, until she was 3 years old. So during that whole time, Debbie had to be a mother and clean up after her daughter, help her get ready in the morning, take her to all of her appointments, while also picking up the role of the father and teaching Franny right from wrong, working a long-hour job so that she can bring money home to provide for Franny, and showing her daughter how she deserves to be treated in life. Finally, Debbie breaks gender norms by organizing a union march at the welding company that she works for. When Debbie fell victim to the pay gap between her and her all-male colleagues, she asked her boss why she got paid so much less, and he said it was because she just constantly had to use the restroom. During her bathroom break, however, she would use the restroom, but she was also breast pumping so that she could feed her baby whenever she got home. To revolt against the company, she organized a union march and led this march, which ended up catching the attention of local news media outlets in Chicago. She was an outspoken, demanding, and adamant person about not returning to work until change was made. Women are constantly told to be seen and not heard, but Debbie breaks this gendered expectation and chooses to be her own person. In this research, we can learn more modern insight into the ever-changing social expectations that women experience, while also revealing the gender norms that most strongly impact women so that these norms can be debunked and changed so that society can evolve as a whole. Thank you.
For good ideas and true innovation, you need human interaction, conflict, argument, and debate. Margaret Heffernan. Hello, my name is Harrison Cook. I'm a junior here at Arkansas State University, currently studying communications, in which I am taking a class called Gender Communication with Dr. Sarah Scott. My presentation today will be over gender expectations within the debate space. As I have a very limited amount of time, what I'm going to do in today's speech is first go over the thesis, go over the gender concepts that I'll be using to analyze my artifacts. I'll talk about my artifacts. I'll also give a little bit of a debate background, and then we'll go into the analysis. So first, looking at gender concepts, what we're going to be looking at specifically is gender expectations for masculine and feminine individuals, performing individuals, specifically looking at 5 different sex. First, with confident and passion. Second, with dressing up. Third, with energy. Fourth, with aggressiveness and courteousness. And 5, with tone of voice. I'll get more into why those are all important here in just a moment. One of the things that we also need to preface this with, in debate, there is a hierarchical structure of power in which the judge is the say-all be-all. Now, with this means that judges can make their decision based on whatever they choose to, and if they have deeply entrenched gendered expectations, that can play a role either consciously or subconsciously into their decision making. What this project aims to do is explore those through the use of ballot analysis, in which judges write their reason for decision and critiques for debaters to better understand the issue. So I'm going to divide up my ballots into 3 different levels. First, at the national level, where judges have the most training and have the most critical understanding. Second is going to be regional, where this is slightly decreased. Judges may be college students, may be hires, may be other individuals who may not have that same level of professionality. And finally, at district, where it's just all hands on deck, we're trying to get as many judges as possible, which means there's not a lot of training that goes on, and analyze the differences. So first, looking at national, looking at all 5 of the different tenets of the ballot analysis, what we found is that at the national level, judges are more professional. The differences were negligible between masculine and feminine performing individuals. Essentially, there were a few instances where it was one or the other, but this could be related to just a simple judge incident. But what we found is that there's not a huge amount of difference at the national level thus far. Second, looking at the regional level at those 5 different, the biggest difference had to do in confidence and passion. So within debate, confidence and passion are typically used as either masculine or feminine terms, with masculine being related to the more powerful confidence adjective versus passionate, which was the more feminine performing adjective. So here we saw that just on confidence and passionate alone, feminine performing individuals were critiqued 9 times versus masculine performing individuals were only critiqued twice. So this is a huge issue in which we are talking about projection of voice, tonality, how aggressive they are in argumentation. This is all goes into kind of the reasoning for this, but I'll go more into my I'll go more in that with my paper. And with confidence, feminine performing individuals were only told 4 times that they were confident versus 5 times that they were passionate, 5 different instances. And most of the time with passionate, it was a critique that they were too passionate. Looking at the second most important thing was tone. Good tone is mostly attributed to masculine performing individuals because of their deeper, richer tones, or considered the better and richer tones, where 2 masculine performing individuals were told that their tone was great versus 0 compliments and or critiques on feminine performing individuals. Now looking at district, which is the behemoth of the project, confidence versus passion. So in confidence alone, there were 2 times in which masculine individuals were told that they were confident and 3 times where if they were feminine were more confident. However, masculine performing individuals had 4 instances of being told to be more confident versus only 1 with a feminine performing individual. This means that confidence is more of a gendered expectation for masculine performing individuals. Looking at dressing up, there were 3 instances in which masculine performing individuals were told that they needed to dress better. And finally, with tone, there were 11 masculine performing individuals versus 4 feminine performing individuals that were told that their tone was great. This is a huge problem. Not only that, but there were 3 feminine performing individuals who got critiques on volumes alone. In conclusion, this paper is starting to find problems in which our gendered expectations affect how we not only interpret the arguments that are being made, but also how we accept them. Something I moved to do with this research project is to highlight this issue and bring it to our attention.
Sexual violence is rarely addressed in military history, particularly during the Korean War. Even rarer, however, is the connection between the defendants' racial backgrounds and sentencing severity. Minimal research has been done on this topic due to the lack of primary sources available, the politicization of issues involved, and the disturbing nature of the crimes in question. The American intervention in foreign countries as a supposed savior in opposition to communism is often treated as a heroic act, but highlighting the dishonorable acts in the wake of this intervention is incredibly important. History must showcase all angles of the actions of the American empire. For every case of rape, murder, and sexual violence in the Korean War, there are countless others that never even made it to court. Using previously unstudied court-martial transcripts, in addition to tracing the ancestral lineage of defendants, this research will delve deeper into the sentencing discrepancy between African-American and white soldiers charged with similar crimes of or relating to sexual violence during the Korean War. The statistics indicate that Black soldiers were consistently punished more severely than white soldiers. Of the death sentence cases, at least 2/3 of them were against African-American defendants. This research explores the individual cases of the soldiers sentenced to death for crimes of sexual violence and uncovers the effect of race on the judicial decisions regarding them. Topics such as sexual violence and racism in the military are uncomfortable for most, but they remain imperative to be discussed truthfully and thoroughly, for without facing the darker side of our own history, we will never see the light. Sexual violence is an unfortunately common crime in the military, but it is also one of the least addressed. The legal system is also no stranger to racial disparity. Between the history of slavery in America and the current state of racial inequality, racism has been a constant presence in the nation. Police violence and racism in both the justice system and the military are present even in today's society, so it's no surprise that the Korean War would exhibit similar, if not worse, statistics in terms of sentencing these defendants. The combination of sexual offenses and racial disparities within the military during the Korean War form the foundation of this research, in which a clear pattern exists. Black soldiers were more likely to be sentenced to death for crimes of sexual violence in the military during the Korean War than white soldiers. The crimes committed were heinous and deserving of severe punishment, but the justice system's delivery of said justice was unequal to the offenders. Of the 43 cases in the database compiled by my mentor, 6 of them closed with a death sentence. Based on the trial records, the cases that resulted in a death sentence were those that included both murder and rape. However, some cases, such as that of U.S. versus Fowler, DeCoster, and Jackson, in which an unnamed Korean female was violently raped and murdered, the defendants did not receive the death sentence. And yes, these 3 men were white soldiers. Of the death sentence cases, however, 4 of the defendants' identities were confirmed and mentioned in official court documents to be Black soldiers. The military court-martial transcript specifically identified these Black soldiers as Negroes, which in itself suggests the presence of racism within the court. Most of the racial identities were confirmed in the primary source court-martial transcripts and confirmed with census data alongside military draft records. I will not describe any of these cases in detail as they are incredibly disturbing crimes, but the following is a brief summary of each case with confirmed racial identities. Private Ernest L. Ransom was hanged in 1957 for the rape of a small girl and the murder of a Korean guard in 1953. He was described as a Negro from Gettysburg, North Carolina in the transcript. The 1951 case of U.S. versus Borner, Howard, and Ware resulted in the death sentence for Sergeant David J. Borner and Sergeant Nathaniel Howard, while Private Clifford E. Ware received life in prison for charges of murder and rape. All three men were Black soldiers. In the case of U.S. versus Day, the court-martial transcript details the brief concern of the civil attorney that it was not a fair trial due to racism toward the defendant, Johnny Day, yet the presence of two, quote, members of the Negro race, end quote, apparently deemed it a fair enough trial. He was charged with murder and rape and sentenced to death. Private Henry L. Hunter of the 512th Engineer Dump Truck Company was a Black soldier sentenced to death for multiple murders and rape charges in 1951. Although the president himself overturned the death sentence, the CMA confirmed it anyway, and Private Hunter was put to death. Upon research into the ancestral lineage, the remaining two cases defendants are unconfirmed, but with multiple potential African-American identities. While researching all of the convicted soldiers, I noticed a consistent pattern in which there were significantly less records of Black soldiers than white. Where I could easily find records and newspaper clippings of births, weddings, and deaths for white soldiers, the only consistent record of Black soldiers was the legally required census and draft cards. I found several potential identities for the U.S. versus Long case that indicate he could have been a Black soldier as well, but at this time I will not say such a thing with full certainty. However, the potential for yet another one of the soldiers sentenced to death to have African ancestry only furthers the claim of racial disparity within these cases. With this in mind, at least 2/3 of the death sentences were set upon Black soldiers. This uneven ratio, combined with the fact that multiple white soldiers were convicted of the same or similar crimes without being sentenced to death, it can be confidently concluded that African American soldiers experienced a severe form of discrimination in the court system for crimes of sexual violence in the military during the Korean War.
Hello everyone, my name is Julian Green, and I want to share with you my research and personal perspective about sonic pollution and its impact on today's music. What is sonic pollution? Sonic pollution can take the form of sounds that consumes the acoustic space and a conglomeration of mess that usually presents itself with negative connotations. These sounds generally are derived from the low-level buzzing oscillations, emergency vehicles passing by, construction noise, and even cell phone notifications. For instance, in 2005, citizens in Tokyo wore earplugs due to the loudspeaker messages and other forms of city noise. This pollution causes disturbances in our acoustic environment and are typically difficult to ignore. Composers are conditioned by the effects of the constant sounds of modern society. Many contemporary composers have been keenly aware of sounds created by society and the natural world. This list of composers have purposely crafted their own expressive compositions that utilizes sonic pollution and natural sounds in unique ways, such as Ryuichi Sakamoto, Clara Iannoda, Peter Valek, Tennyson, Mid-Air Thief, and Adam Neely. Next up is a few examples of compositions by some of these composers. Here is Energy Flow by Uuzan and Ryuichi Sakamoto. In this piece, Ryuichi Sakamoto uses pre-recorded sonic sound waves and organically gestural techniques in tandem with a more pop-like sound. In Clara Iannoda's Dead Wasp in the Jam Jar, she focuses intently on how sound behaves when sustained and molds this into a cohesive manner. In Improvisation for Prepared Acoustic Guitar and Prepared Refrigerator, Peter Fallick encapsulates the improvisatory elements through more percussive means. Here is a well-crafted popular piece by Tennyson called Like What that utilizes natural sounds and man-made tones.
One moment, please.
Here is a segment of my composition in the making that utilizes abstract sounds in unconventional ways. This gesture here brings out a more noisy and obtrusive quality in the sound produced by the saxophone. The tam-tam further enhances the noise aspect of the piece by being played unconventionally along the outer rim in a similar gesture such as the saxophone. The guitar acts much more similarly as the saxophone, except the pitch bending is much wider in comparison. Here, the pianist is playing these percussive notes to demonstrate more contrast in the overall sound of the ensemble. Sound does not always have to be perceived as an obnoxious noise floating around our acoustic airspace. If we can look at sound in a more constructive manner, the musicians can express their values and concerns about society through a more forward-looking perspective. With this newly garnered perspective, we as a society can take a better look at what sonic pollution means to us and how we can utilize sound more creatively while creating a better impact on today's music. Thank you for your time.
First, I'm going to explain the text that I analyze. Secondly, I'm going to discuss my theory and methodology. Thirdly, my analysis. And finally, I will provide some final thoughts. Bridgerton is a hit Netflix series based on a series of novels. The show is set in the 1800s in England, a time of debutantes and extravagant balls. Eloise Bridgerton, a fictional member of a wealthy family, exhibits rebellious behavior and an unwillingness to participate in society's standards for her as a woman. I believe this media to be important for gender studies because it demonstrates the societal expectations for women during this time and how women began to break the norm, even in small ways. I textually analyze this media to provide a theory. The objectification and dehumanization of women, specifically in the 1800s, has created a strong foundation for the gender roles and expectations for women. The specific gender concept I will be focusing on for this presentation is objectification. Objectification can be defined as the degrading of a person to the status of an object. Throughout the series, Eloise Bridgerton protests the expectations of her and her sisters regarding marriage, motherhood, and education. In one conversation with a close friend, Eloise comments on an art display by saying it was quite dull because it, like all these paintings, was done by a man who sees women as a decorative object. Eloise makes a bold statement in a public setting about the male perception of women and their role in society. In this time, women were expected to exist for their husbands and him alone. Young girls often spent their time in this show choosing fabrics for dresses and preparing to meet suitors for marriage. Young women in this show are taught from a young age on how to act and appear in order to attract a husband. This objectifies women by diminishing their individual thoughts and desires and magnifying their usefulness for men. In another public conversation, Eloise expresses her frustration with her mother's obsession to raise her children for marriage and motherhood. She states, "I shall have to stand by and watch dear Mama appear proud because some man should like to admire my sister's face and hair and fill her up with babies." The mindset that women were bred to marry and then produce heirs to continue the family line demonstrates the objectification of women during this time. Women's bodies were seen as factories for children rather than living, breathing humans with emotions and desires. Eloise's mother demonstrates an urgency to marry off all of her daughters in order to ensure that they will be taken care of later on in life. I believe that this is due to the fact that women were not given the opportunity to provide for themselves, not necessarily that their mothers did not want them to do so. Along with being raised for marriage, women were not given the same opportunity for education as men were. Instead, women were taught household skills and etiquette. Eloise also exhibits a desire for more in life by saying, "Having a nice face and pleasant hair is not an accomplishment. Do you know what is an accomplishment? Attending university." By societal standards, only allowing men to obtain college education and not women, women are forced to become dependent on men for survival. This practice only encouraged the objectification of women further by systematically excluding women from the educational scene and forcing them to marry. Women did not begin to obtain college degrees in England until almost 1900, proving that the objectification of women is long-lasting. Restricting women to the role of wife and mother denies women the right to their own thoughts and desires. Eloise Bridgerton expresses her dissatisfaction with the expectations for her as a woman by asking the question, "Why must our only options be to squawk and settle or never leave the nest? What if I want to fly?" Unlike her friends and family, Eloise desires for more in her life than marriage and motherhood. Women during this time period really only had two options, either to marry and have children or remain with their parents until you do so. Eloise sees more options for her future and expresses her want to explore them. She strongly protests the standards that her society has set in place for her by initiating rebellious conversations. While this may seem like a small act, the fight against objectification had to start somewhere. In the Netflix series Bridgerton, Eloise Bridgerton rebels against societal norms for women in the fight against objectification. She inspires viewers to break norms and demonstrates the impact that this has on society. This is interesting because even though that the ways women were objectified in the 1800s may seem extreme, objectification still occurs in our society in very obvious ways. Thank you.
Hello, my name is Rachel Haywood, and I am a junior here at A-State. And this is my analysis of self-identity and social norms in the film Moonlight. At some point, you got to decide for yourself who you're going to be. This was some advice given to a young boy named Chiron in the 2016 film Moonlight. It chronicles Chiron's struggle to find his self-identity as a poor Black gay male from childhood to adulthood. Throughout the film, Chiron battles social pressures to meet society's standards of masculinity, while at the same time moving further and further away from the person he truly wanted to be. The textual analysis that I'm going to conduct is crucial in understanding the internal and external struggles that Black men and boys face when dealing with the effects of social norms when crossed with intersectionality and self-identity. I decided to conduct this research on this topic because I felt as if the first thing that comes to people's mind when they hear the term gender are women. And I wanted to shine a light and direct the focus of this project towards men, and in this case, poor gay Black men, as it is very important to the study of gender overall. Okay, the thesis in gender concepts I have constructed is, while this is an ongoing research project, the preliminary findings suggest that our sense of self-identity is directly linked to how others perceive us to be. And gender concepts I will be focusing on, but my main focus will be identity construction. But to back that up, I'll have intersectionality mainly. Okay, so self-identity is defined as the way that people see themselves as individuals and as members of groups, as well as how others see them as individuals and members of groups. Identity includes, but it's not limited to, gender, sex, race, class, sexuality, and nationality. When these identities cross with one another, we are along the guidelines of intersectionality as proposed by scholar and race feminist Adrienne Wing, who explains that identity is multiplicative rather than additive. And in this case, Chiron's intersectionality consists of being Black, gay, poor, and male. In the film, one of the first people that tries to identify Chiron is a character named Juan, who ultimately becomes Chiron's father figure, as Chiron is being poorly raised by a single mother. And the advice that was given to Chiron is, at some point, you got to decide for yourself who you're going to be. You can't let anyone decide that for you. The next character that helps to construct Chiron's identity is a character named Kevin, who was a childhood friend of Chiron and through his teenage years also. Kevin was the only kid when they were younger that actually took the time to get to know Chiron before judging and bullying him like the other kids. He gives Chiron the advice to try his best to show the other boys that he is not soft, so they won't pick on him. And lastly, the final character that shaped Chiron's identity was his mother, who actually denied him the love that he needed as a child, which was partially and ultimately the cause for Chiron's hard exterior as an adult, and him resulting to becoming like the only father figure he known, which was Juan. And in conclusion, the research that I conducted highlighted the masculine standards that Black men and boys are expected to uphold, along with internal and external repercussions that form when they fail to do so. And also, it concludes my thesis statement that other people's perception is basically how we as individuals form our self-construct and identity. And thank you.
Hi everybody, welcome to Constructing and Editing the Digital Filters for She Kills Monsters. I was the filter engineer for our Zoom production of She Kills Monsters Digital Realms, which was back in October. I just wanted to go through and discuss some of our filters that I worked on. The best part about doing filters as opposed to doing actual props or costumes is that it's a lot easier for the actors. They don't have to worry about so many different aspects of their characters and what they're going to be doing. So, for example, Agnes, she was our main character, and she didn't have a very big filter. She only had one that came into use at the end. What it did is it added the sparkle effect for her. It gave her some shinier eyes, and it gave her a nice pink background that would follow her as she moved. So it took her from being a person who was playing D&D sitting at a table to someone who actually believed in the world and someone who actually was a part of the magical realm, because all of the other characters she'd been playing with who were part of the D&D session all had filters that denoted whether or not they were part of it. So she had this at the end: sparkly eyes, the pink box, or the pink background, including the little pink box, and the bokeh is what we call it. So the sparkles. We used the bokeh a lot because we really liked how it looked. That was Agnes. Hers was very simple. Going into one that's a little less simple, Steve, who is one of the mages, he was one of the other D&D players who would appear for a couple of minutes and be, a mage. He was wizard. He used magic. And so we gave him this funky hat. We gave him a lot of sparkles. I believe he's got the shiny eyes on as well. So you can see when he's actually facing the camera, the hat is following his head. And if he tilts it a little, I don't know that this actor is going to. It's not the greatest because he's so far back, but it will follow his head. It will move as if it were an actual hat. Which was a really fun aspect because he had a lot of quick changes that were about maybe 20 seconds. He would leave the scene, I suppose, and then he would come back on, and he would have to have this hat and then this robe, and it was just a very, very fun time. And then we had some characters that were like Farrah Fairy. Hers was very difficult to work on. She had wings, she had sparkles, she had some makeup on. She was very much a transformed person. As far as physical things that we gave her, it was pretty much exclusively a dress. We didn't worry about anything else for her costume. We gave her wings, we gave her bright pink makeup, we gave her all these little sparkles of bokeh that are around, and we edited the bokeh so it wasn't quite so floaty. We wanted something more intense and more sparkly. So if we go back to the person who was dancing, we should be able to see it a little better. There, you can see it a little there. So she was about this far back. She had the wings, they would follow her. They looked like they were sitting on her shoulders. If she turned her head, they wouldn't follow her head. And you could definitely tell that she was supposed to be a different person. And so all of these quick changes, things that they needed to get into very quickly, things that had they been physical would have hindered their performance, they didn't have to worry about. We made it all digital for them. So they came, they did their part, and we did our part on the digital end to just make them look amazing.
Hello, my name is Kiri McMahon, and I will be presenting on a project where I created an oboe performance version of a Baroque violin sonata. In the Baroque period, it was customary for composers to use only sparse notation in their sheet music, which left much more to the performer's discretion. Ornamentation and articulation, in particular, were often either implied by the musical structure or simply left to the performer's preference. Therefore, if a contemporary musician were to perform a Baroque sonata by only incorporating the notation on the page, it would lack half the nuance. For my project, I compiled an oboe version of a Handel violin sonata, HWV 373, number 15, with my own articulation and ornamentation. It was based upon several different editions, including the first published edition, Albert J. Andrews' Transcription for Oboe, and Henle's Urtext edition, edited by Stanley Sadie. It would be impossible to condense my entire project into a 5-minute presentation, so I will explain what I did in a small section of the second movement to convey a sense of my process. Here are two of the editions I referenced. On top is the first edition, with figured basses notation for keyboard accompaniment, and in the middle is the Andrew oboe transcription, transposed into F major to account for the change in instrumental range. This last staff is my version, which is also transposed into F major, but which had no articulation at this stage. It sounds like this. This is currently a bit monotonous, especially considering the unchanging rhythmic pattern of 16th notes, so I decided to add some slurs to give it a more clarified structure, starting by looking at what the other editions do. In the first edition, the first 3 16th notes of every other measure are slurred. Handel presumably wrote these in himself, and one of my goals was to honor Handel's compositional intent, so I decided to incorporate them into my version. This is what it sounds like. However, Andrew discards Handel's articulation and instead slurs the last 3 16th notes in each measure. I was less inclined to trust Andrew than Handel, so before I decided whether or not to adopt his slurs, I considered the musical structure. These are the different structural ideas highlighted in different colors. The orange sections are the last 3 16th notes in each measure, the ones Andrew slurs, which follow a pattern of going down to the next note in the scale and then back up, like an inverted mordent. The one circled in orange is the same pattern but backwards, going up to the next note instead of down. These other colors, blue and green, are also analogous parts. All of them are the first 5/16 notes in a measure, and they are all a scalar ascent followed by a descent. The difference is that the blue sections have a 3-note ascension and descend in thirds, whereas the green sections have a 2-note ascension and descend by the scale. Handle slurs are on the first 3 notes of each blue section, which is logical because they comprise this ascension. However, I decided to slur the first 3 notes of the green sections as well, which emphasizes their similarity to the blue sections. Here's what it sounds like. In addition, there is another important structural detail in the accompaniment to consider. Looking at the figured bass, there's a rhythmic pattern of an 8th note followed by a quarter note, which emphasizes the first and second to last 16th notes of each measure. This led me to think that they are the most important notes in each measure, and that gave me an idea for how to consider Andre's slur. I decided to try slurring the last 2 16th notes in each measure instead of the last 3 like he does, because that'll put an extra emphasis on the second to last note. Here's what it sounds like. This does indeed provide an extra emphasis, but upon listening to it, it becomes clear that it breaks the orange section into 1 note followed by 2 notes. That must be incorrect because as a section sharing this distinct pattern, they belong together. Therefore, on second thought, I decided to use Andre's slur and create the extra emphasis using only my air as I play. This is how it sounds. That concludes my demonstration of the sort of process I used to create my oboe performance version of this sonata. I'd like to thank my mentor, Dr. Letterman, for all her help with my project and presentation, and my viewers should know that I greatly appreciate them watching. Thank you again.
My name is Jay Riggs, and my presentation is Sexual Abuse of Boy Scouts Inflicted by Members of the United States Military During the Cold War. Not long after World War II, the United States found itself in another conflict with the Soviet Union. Fear of communism was not new in the United States, but the threat of a worldwide communist campaign led the United States to intervene. In result, the Cold War began. Although no direct fighting occurred, the two nations engaged in an arms race and sought to antagonize each other. Although communism was perceived as a detrimental threat to America, there was a pressing domestic issue that desperately needed to be addressed: child sexual abuse, specifically child abuse inflicted by servicemen of the United States military. Between 1950 and 1970, there were multiple instances of servicemen abusing their positions to molest young boys involved in one of the most patriotic youth groups in the United States, the Boy Scouts. Members of the military, regardless of rank, were able to volunteer as scoutmasters of troops. Scoutmasters had a position of authority and were responsible for the boys. They would act as teachers and mentors to the young scouts. Although the position seems innocent, some servicemen took advantage of their position and sexually abused children. After studying 6 servicemen accused of sexually assaulting Boy Scouts, all under age 16, it became clear that much more came into play than the crime itself when sentencing members of the military. Factors such as rank, behavior, and even sexuality came into play. Of course, those accused of abusing multiple children received harsher sentences, but their character and contribution to the military was still considered during the trial. In 1952, Major George A. Dunn of the United States Air Force was accused of committing lewd and indecent acts and sodomy per annum as alleged upon males under 16 years of age. The scouts' age ranged from 11 to 14 years old. Dunn, acting as a scoutmaster when he committed the act, sexually abused the children to benefit their health. Experts and psychiatrists disputed Dunn's sanity during the trial and while he committed the heinous acts. One psychiatrist claimed that he could not determine right or wrong when he committed the crime. The same psychiatrist diagnosed Dunn with developing schizophrenia and as a sexual deviant. Other psychiatrists also agreed that Dunn was a sexual deviant manifested by homosexuality. Dunn's diagnosis became a key factor considered in his trial. Aside from the homophobic social climate of the Cold War and homosexuality still being perceived as a mental illness during the 1950s, Dunn being diagnosed as a sexual deviant with homosexual tendencies potentially made the court more likely to find him guilty. Homosexuals were assumed to be more likely to molest children than heterosexuals. Initially, Dunn was sentenced to dismissal and 30 years of confinement and hard labor. Upon review, it was reduced to 15 years of confinement. In 1953, third-class airman Donald M. Bond of the Air Force was charged with sodomy, but the court instead convicted him of committing indecent and lewd acts by placing his hands upon the private parts of a young Boy Scout. The incident occurred after the Boy Scouts began swimming in the nude and initiating an obscene game. Bond attempted to end the game, but eventually grabbed the Boy Scout and committed the assault. However, due to the absence of instructions upon the offense and indecent acts with the child, eliminated any possibility that the act found could be equated to that offense. Essentially, the lack of sodomy significantly lessened Bond's sentence. Bond's sentence was also lighter due to his youth, it being his first offense, and quality of military service, along with his attitude and loyalty to the military. The Board of Review ruled for Bond to serve 1 year of confinement at hard labor. In 1967, just 12 years after Bond's light sentence, Captain Robert H. Bruskin, DDS of the United States Army, was accused of sexual assault with intent to commit sodomy upon a child as a non-consensual offense. Bruskin attempted sodomy with 2 minor Boy Scouts aged 13 and 14 while working as a scoutmaster. In both instances, Bruskin began by wrestling with the boys before pinning them and engaged in lewd acts. Both victims gave their testimonies to which Bruskin verified aside from the acts of indecency. He admitted to the wrestling, but never to the acts of sodomy. Bruskin's character and background came into consideration during his trial. He was praised for his military and academic performance. 16 witnesses testified to his integrity, character, skills, and religious faith. Petitions were even signed for Bruskin to have a lighter sentence. With an abundance of support for Bruskin's innocence, his sentence was reduced from 18 months to only 1 year hard labor. These cases expose hidden crimes of members of the United States military during the Cold War. Although all were found guilty and served at least 1 year of confinement, all initial sentences were reduced, showing a leniency for servicemen regardless of crime. Reduced sentences also show a lack of justice for children sexually abused by men that were supposed to mentor and protect them. Cases such as U.S. versus Bruskin and U.S. versus Bond reveal a flaw in the military justice system. In both cases, the character and performance of Bruskin and Bond is taken into consideration despite the accusations. Bruskin's amazing reputation and ability to conjure a plethora of witnesses led his already light sentence to be reduced by 6 months. Bond's youth and ambition for military service also allowed him to get a much lighter sentence. Reducing these sentences also provides a sense of protection for well-performing servicemen. The better the reputation, the lower the sentence. Most importantly, all these trials reveal a threat during the Cold War much more insidious than communism: sexual abuse of children. Child sexual abuse scandals were well hidden in the Boy Scouts for years. These trials provide insight on how servicemen use their authority and position to take sexual advantage of children and how they were punished.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hello, my name is Jackie Zell, and I am a music composition major here at Arkansas State University. After I get my composition degree here, I plan on getting a music technology or production degree from another school. I hope to one day be a freelance composer and producer or to work in a recording studio. I'm going to be discussing my piece, Fog on the Hive Mind, and how it pertains to the language of music and using that language to express the human experience. Music is the manipulation of pitch, resonance, rhythm, timbre, texture, and space. This broad spectrum of manipulation gives music a unique opportunity as a language to communicate human expression in very precise ways. Music is remarkable in this way because, due to the abstract nature of music, the expression it projects defies typical human bias and stigma. To truly understand the language and to be able to express myself in the most precise way, I needed to develop my understanding of the details. Using music software like Ableton, I am able to alter every single aspect of any sound and manipulate my music to sound however I want it. I composed the piece Fog on the Hive Mind with this idea in mind and used it to explore these aspects of music: resonance, texture, and timbre. The two sound sources I used for this piece were a guitar and a kalimba, and both of those instruments are known for their resonance. I was able to manipulate the sound by isolating certain frequencies created by these resonances and changing those frequencies to create a variation in texture over time. I also experimented with certain digital effects and created sounds from these instruments that sounded nothing like the original source. I used this to my advantage and created a variation of timbre. The aspects of pitch and rhythm were slightly random for this piece, and it started out with me playing an arpeggio that I really liked on kalimba and playing obnoxiously difficult polyrhythms on the guitar that matched the kalimba in pitch. This combination of ideas created the framework for this composition and displays the innate ability to drastically alter the sound through other aspects besides pitch and rhythm, those being resonance, texture, and timbre. The system I used to write this piece was heavily based on experimentation. Even the original sound sources were ideas that I came up with on the spot. I would try out different digital effects or playback speeds to see which one most closely matched what I wanted to hear, and I experimented further with these digital effects to figure out how the effect can manipulate the sound over time. Another part of my system for this piece was repetition. Through creating repetitious ideas that change in texture and timbre over time, I can bring the listener into a sort of trance, and then I can wake them up from the trance with something completely different. This idea of a trance comes from my distaste of modern communication and media. We are force-fed information, whether it's true or not, every day of our life since we are born. And being able to break free of that loop of constantly needing information is represented by a drastic change in all aspects of the music. The final feature of my system was dedicated to the most ornate details. I spent hours trying to figure out how loud I wanted a certain voice to be in a certain section, or how much I wanted to increase distortion on my guitar track, or how long I wanted a certain voice to go on for in the music. This last step of my system is what I believe to be the most important, and it ties into the idea that music lets me express myself with remarkable accuracy, and that the studying the details truly gives the music I create the most meaning. I will now play a selection of this piece to display the topics covered in this discussion. Thank you for your interest in my creative state presentation.
Shelley Gibson and I have used our creative habits and ideologies to make a print that can relay the strength it takes to succeed in this world. After deciding our message, Shelley disguised and photographed a model specifically chosen to add contextual meaning in various poses. We decided together which pose best translated a character who could brave anything. We were looking for an image that embodied defiance and aggression, love and solitude, strife and survival, and honesty and resistance. The figure we chose is full of unresolved feelings that are just out of reach.
We then chose a call to action word, resist. We chose that word because it invokes thoughts and ideas about power struggles, both internal and external. Such words describe an event, playing with the history and connotation of language. Geographic and historical use and the roots of language develop additional layers of meaning. The character of the word is reflected in the figure because the figure is capable of making a stand, to withstand and refrain. The look of indignation on the model is obscured by the darkness of the eyes, creating an ambiguity because of the timid chin. The figure transcends place with sculptural hair and a powdered body. She is a strong female character, like those in literature and popular culture, and references such sources as historical murders, film noir, ceremonial sculpture, and war photography.
So in activist art, ideas are written and pictured in ways to get ideas across to the public. Alongside the posters, videos, and other means of advertisement are the voices and minds behind the matrix. The idea of art and voice working side by side is a concept I've been experimenting with. In the past, I found a button that said, "Eat Right America" in a thrift store. The statement was there, but the voice and information behind it was not. My favorite part about finding this button was the realization that its vague text left without explanation was an opportunity for me to make it mean anything I wanted. And also anyone else that would make it mean what they wanted. So my goal in activist art would be to make prints that I could stand behind and explain, while also making it a resource for others to use and speak on their own beliefs too. So in the artwork, I use repeating text and sometimes illegible text to put an emphasis on things. And I kind of learned this practice through graffiti. So any artist or writer has one name that they go by, and it means everything that they believe in. So that's kind of what I picked up, but I just put the word out there so everyone could know and everyone could talk about it.
Okay. After this, we could begin the process of experimenting. So we chose different colors and ways to layer the script and the image together. We put them in between and over the top of each other. We combined different layers and divided colors, creating textural surfaces, playing around with image degradation and fragmentation, and then moved the image beyond its photographic origins. These layers are both physical and emotional veils.
So our intention with this piece was to theatrically show the vigor one must carry within to resist the injustice in the United States and fight for yourself and what is ethically right for others. Shelley and I felt the need to remind anonymous viewers, friends, and family that although the world caught into flames over the last years, that everybody can still fight for what they believe in.
We're pretty excited. This print, it will be part of the upcoming 2022 Mid-America Print Council Conference. It is an exchange portfolio called Open-Ended CTA, a collaboration. There's an edition of 31 prints, and they'll be exhibited and become part of permanent collections. Thank you.
Hello, my name is Samuel Scott. I am a senior theater major, and I have created an original electronic music piece that alongside Dr. Timothy Chris at the Department of Music. My piece is called Get Hot, and it utilizes the technique of sampling. Sampling in this case is taking parts of a song or a previous composition or an audio recording and then repurposing it into a brand new composition. So why I decided to do this? Well, I decided to do this as a way of exploring experience from a previously familiar experience. So sampling in this case is similar to using quotes or excerpts of text from one author into another written work. The purpose of it is to serve as a reference of the previous original work and all the experiences that you have, and then trying to create a brand new experience, basically through manipulation, through recontextualization, and then just building a brand new experience out of that. It uses to reinforce brand new ideas from it. This is popular in many other art movements, an example of which is the pop art movement, which has Andy Warhol and Roy Lichtenstein in it, where you take previous pop culture references in like Campbell soup can and then recontextualizing it into a high art aesthetic. And this is why I decided to do with my piece. So my piece, I use 3 samples. I use Steam Heat from the 1954 musical The Pajama Game. I use another song called Fragments of Crystal by Italian film composer Giovanni Cristini. And I use my third sample is a song called Face from experimental hip hop trio Clipping. So the main sample of it is Steam Heat, and how I discovered it, well, it relates to my field of theater. And I have listened to this song at one point in my life, and what led me up to building a new song from it is, I was listening to it, and then at one point, all the lavish instrumentation just breaks down, and then all of a sudden it's just clapping, stomping, and singing, and with different phrases. And in this case, Get Hot is where the title comes from. And I thought this is interesting, and it reminded me a lot of industrial music and experimental music. And I decided to experiment that and then go down that route and then actually build an industrial dance music out of it. And the songs, Fragments of Crystal and Face that I use as my samples, I used it to reinforce those aesthetics, to reinforce that sort of mood that I was going for. So what I did, I basically took those samples, I chopped them up, I pitch shifted them, I time stretched them, and I added effects to them like reverb and some delays. Now, I chopped them up into multiple different patterns, I put them into different scenes, and then I put them into different sections and I added them with multiple different sections into the song, so that way it becomes a lot more dynamic. To reinforce the industrial dance aesthetic, I decided to add a kick drums and a heavy bass line to it, so that way it adds to the aesthetic. And for more experimentation, I decided to use synthesizers as background texture. There's even a section at one point where all the instrumentation just breaks down and it's just all the synthesizers and one of my samples, which is actually Fragments of Crystal, which I have slowed down and reversed. So sampling to me is a way of just building experience. It is something to take the moment that you have experienced and create something entirely of its own that's something brand new. So I would like to share a little bit of my work, a little bit of what I have made. So this is my piece right here. This is my piece. Now I will put in a full link so that way you get to listen to it yourself. So here's a little bit of it. Anyway, this is my presentation. Thank you so much for taking the time to watch my presentation, and I hope you have a great day. Thank you.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
Hey everyone, my name is Matthew Emery, and I am here to tell you guys a little bit about my paper. It's titled A Historical Look at Distrust in Contemporary Media. And that title's a little self-explanatory, honestly, right? It's we're looking at contemporary media, so the media we have today, and I guess one thing to elaborate on is the media part. I guess by that I mean the American news media, the journalist, right? We're looking at the current state of that because in my opinion, it is not in a healthy state. And I think a lot of people would agree with me there, right? I think in the last few years, we've seen this rise of fake news, right? These allegations and in some instances truths, right? Where just about everybody in America knows or has heard of the term fake news, right? It's haunting journalism right now. And I think 2020 was such a culmination of everything in a sense. I think the US presidential election had a huge influence over how Americans perceive media, and specifically the distrust they have for the American news media. And I think the COVID-19 pandemic played into that as well, with all this disinformation, this fake news, just people just reluctant, hesitant to believe things, right? So I believe that there's this rising distrust in our news media right now. What my paper does is I am drawing an analog to where we're at right now to the 1940s in America. And you may be asking, why the 1940s? What's so interesting about the 40s? In the 40s, in my opinion, and what my paper goes on to talk about is, well, we were actually in a very similar spot in the 40s that we're in right now when it comes to American news media. In the 40s, there was a boom of new technology that left journalism in a very sensitive spot, right? And then came, practices like yellow journalism, which just destroyed the trust that the American citizens had in the press. So much so that a lot of people felt like the First Amendment was at risk. And so to combat that, these intellectuals at the time out of the University of Chicago, they formed a group that would be known as the Hutchins Commission, and they laid the bedrock for American journalism for years to come. And they did this as a way to secure and protect the First Amendment freedoms that the press enjoys here in America. And ultimately, that's what my paper is about, is drawing this analog between the 40s and now. Similarities, like the similar problems they faced that we're facing, and specifically, what did the commission do to address those issues and regain that trust in the American citizens? And that's what we're doing with my paper. We're looking at the issues that are causing this distrust currently. This distrust has been documented by the Edelman Barometer, the Harris Poll. It's very clear that the American citizens do not trust American media right now. So we are trying to analyze, okay, we know that this is the situation we're in. What can we do to rectify and earn back the trust of the American people? And we do that by looking at the Hutchins Commissions from the 40s. And the ultimate conclusion that I draw in the paper is that we need a Hutchins Commission 2.0 to form and lay a new groundwork, a new bedrock for journalism, something to get us through this digital age that not even the Hutchins Commission could have predicted. We need a new bedrock to carry us through the digital age and beyond, and restore our trust with the American people and protect the First Amendment for the press.
Hi, my name is Phoebe Haynes, and I will be presenting the historic landscape of Mildred Jackson Elementary High School. Share my screen. The historic landscape that will be discussed will be the Mildred Jackson Elementary High School, presented by me. Why is this important? This was a predominantly African-American school in the 40s through the 60s in the rural town of Hughes, Arkansas. These African-American students were forced to deal with systematic racism, segregation, integration, hard labor, as far as chopping cotton and picking cotton, while trying to pursue an education. My mother, who is an alumna of Mildred Jackson Elementary High School, her name is Conster Hines. I felt it was important for the world to know about the personal stories from both the alumni and the alumnus' experiences at Mildred Jackson and how they overcame those stumbling blocks and earned their high school diploma. The methodology that went into this research was in-person and over-the-phone interviews from a mixture of both male and female participants. A total number of 7 participants that included 1 male and 6 females. 3 in-person interviews and 4 over-the-phone interviews. Those interviews lasted between 30 minutes to an hour and a half. The interviews lasted between June 2020 and July 2020. My goal initially was to interview 20 participants, but with the heighten of COVID-19, people didn't have the openness to participate in my research, which is totally understandable. The findings. The impact of the core values that my mother and her peers learned while at Mildred Jackson Elementary High School. The struggles with their hardships of dealing with systematic racism in their school, as far as the unpleasant temperatures and the dilapidated buildings, the hand-me-down textbooks from white children that were used, written in pages that were also torn out, and having to leave school early to work in the cotton fields until sundown, or being absent from school months at a time to work in those cotton fields. And they also earn their diploma. Future research. I would like to revisit the participants that initially participated in this research to see if they have any more that they would like to add to their personal stories. I would like to also research the white student who attended the predominantly white school of Hughes High before and after integration, and get their experiences on what they felt when their school was integrated, what it was like living during segregation in the segregation era, and how they felt seeing the African American kids in the cotton fields when they should have been in school too. Next will be direct quotes from the alumni and alumnas experiences when they attended Mildred Jackson Elementary School and what Mildred Jackson means to them. The song that's playing is Up Against the Wind by Lori Perry.
Seems like I push on they just keep knocking at the sun. It seems since I was born, I've wondered every morning down on my luck and the is wind. It's not over.
Thank you for watching.
Hello, my name is Luke Parker, and the title of my project is Grant Professionals Finding a Home in Public Administration, an analysis of the need for a grants management concentration in Masters of Public Administration programs. Dr. Donna Hanley is my mentor for this project, which is proposed as a thesis topic for the completion of my Master's in Public Administration at Arkansas State University. The purpose of this presentation is to discuss some of my findings from the initial literature review phase of this research project to initiate discourse on the topic with the Arkansas State community. To start things off, I want to provide a brief background on grants management. But what are grants? To quote grants.gov, grants are, quote, the way government funds your ideas and projects to provide public services and stimulate the economy, end quote. Not all grants are governmental. Indeed, there are also corporate and foundation grant makers. However, federal grants to state and local governments, otherwise known as intergovernmental grants, make up the majority. According to data from the Foundation Center and the Office of Management and Budget from 2015, the most recent year for which data is available for comparison, there were over 10 times as many intergovernmental grants as there were grants from other non-governmental sources in 2015. Together, these figures represent nearly $700 billion in grants from over 86,000 corporate and foundation and 26 federal grant-making organizations. Not only is that a lot of money going out, but it's a lot coming in, which is where the role of grant professionals becomes ever so important. There were over 42,000 recipients of discretionary and block grants in the federal government's fiscal year 2021, which means 42,000 subnational government entities needed personnel to manage these grants, and likely others too. While the Bureau of Labor Statistics does not collect data on grant professionals as an employment category, the categories of fundraiser and public relations and fundraising managers have been used as anecdotal data to indicate the number of professionals in the field, which equates to just over 160,000. This lack of data regarding grant professionals is important to note, however, as it hints at the main issue I'm going to be talking about, which is the lack of scholarship about grants management as a profession. So searching library databases using grants as a key term yields many results, most of which deal with the technical writing concepts used in developing grant proposals. Refining our search to public administration journals, we see a growing trend in scholars critiquing problems with intergovernmental grants while also acknowledging their relevance to public policy. We can also see this culminating in national politics, such as in the 2018 hearing before the House of Representatives Subcommittee on Intergovernmental Affairs and the subject of federal grant management, in which Representative Gary Palmer, the chairman of the subcommittee, stated, quote, annually the federal government loses track of millions of dollars due to a failure to review and reconcile grantee reports in a timely manner. It is my hope that this Congress can continue to help this effort with continued oversight of the grant-making process, end quote. This hearing was also important because it included witnesses from 5 federal agencies to discuss methods and needs to streamline intergovernmental grant systems. With such emphasis on improving grants management, the elevation of the grant professional seems warranted as well. Currently, the Grant Professionals Association and Certification Institute offers the only credentialing process awarded through psychometric evaluation of core competencies, which we'll discuss in a moment, and is accredited by a third-party accreditation entity, the National Commission on Certifying Agencies. The organization administers its own academic journal, in which dozens of articles and strategy papers can be found relating to this very subject of grants management education, particularly the need for a degree conferring program in which, in addition to the certified grant professional credential, to fully professionalize the field. An attempt to find these articles through Arkansas State University's library system yields no results, however, which further signifies the professionals in the field are attempting to find a home within a deeper epistemological purpose, i.e. grants management as the epistemology and public administration is the deeper concept. In other words, grants management degree programs do not yet exist because the concepts in the field have yet to be connected with the larger, deeper school of thought. Looking at the GPCI core competencies as what would be required for a professional in the field, we can determine they complement the MPA curriculum. Competency 1 states knowledge of how to research, identify, and match funding resources to meet specific needs. Competency number 2 is knowledge of organizational development as it pertains to grant seeking. Competency number 3 is knowledge of strategies for effective program and project design and development. Competency number 4 is knowledge of how to craft, construct, and submit effective grant applications. Competency number 5 is knowledge of post-award grant management practices sufficient to inform effective grant design. And competency number 6 is knowledge of nationally recognized standards of ethical practice by grant developers. Competency number 7 is knowledge of practices and services that raise the level of professionalism of grant developers. Competency number 8 is knowledge of methods and strategies that cultivate and maintain relationships between fund-seeking and recipient organizations and funders. Competency number 9 is the ability to write a convincing case for funding. So to wrap this all up and really describe what this project is about, I wanted to briefly note that my research methods for determining the need for a grants management concentration includes doing content analysis of accredited MPA programs to determine the extent to which they align with the GPCI core competencies. And in doing so, my hope is to produce results that can be used by MPA faculty to determine the need for a concentration in grants management. Understanding that a qualitative investigation such as this requires stakeholder input, I also hope that this presentation will serve as a springboard for larger conversations with the Arkansas State community to help determine if a concentration is needed and what the potential impact could be of that. I appreciate everyone's time and consideration in listening to me talk about this subject. And thank you and have a great rest of your day.
Some presentations are the results of a classroom project, oral history, service training, assessment, journalism, or quality improvement activities. Such activities may have been deemed not to meet HHS OHRP definitions of human subject research designed to develop or contribute to generalizable knowledge and thus did not undergo IRB review.
I am Shobhitan Kandasamy, and this is a presentation of my submission of the nomination of Historic Dice Colony to the Historic American Landscape Surveys. The mission of the Historic American Landscape Survey, referred as HALS, is to record historic landscapes in the United States and its territories through measured drawings and interpretive drawings, written histories, photographs. The National Park Service oversees the daily operation of HALS. Historic landscapes are special places that are important touchstones of national, regional, and local identity that foster a sense of community and place. Historic landscapes are also fragile places. They are affected by forces of nature, development, vandalism, and neglect. They undergo changes that are often unpredictable and irreversible. For these reasons, and for the benefit of future generations, it is important to document these places. This presentation is organized based on the HALS guidelines. The nomination was submitted in summer 2020. Dicetown is bounded by ditch number 40B to the north, Tyronza River by west, and the County Road 956 to the south. The farm number 266 house is located to the west of Dicetown. So why is Dice, Arkansas significant? DICE was founded by the federal government in 1934 in a New Deal program during the Great Depression. This agricultural resettlement colony has national significance as the largest agrarian community building project that was created as a national sociological experiment in rural rehabilitation. DICE Colony Center, pictured here in an early drawing plan, was also referred to as the community center and is listed as a district in the National Register of Historic Places for its significance in modern American social and agricultural history. DICE also contains the Farm Number 266 house, the boyhood home of singer-songwriter J.R. Johnny Cash. This house has statewide significance in its influence of Johnny Cash's musical career, recounting his childhood in DICE, as well as local significance as a Federal Emergency Relief Administration constructed residence. DICE is a Mississippi County in a flat, fertile strip of land west of the Mississippi River in the northeast corner of Arkansas in a region called the Arkansas Delta. Between 1934 and 1939, Colonization Project Number 1, renamed as DICE Colony, was constructed on 16,000 acres of swampland that was cleared to create a planned rural community and contained 500 homes with an adjoining farmland. The landscape also included the DICE Colony Center, pictured here, which consisted of an administration building, a hospital, a town center, and other small town facilities. The landscape has evolved through the years with new structures, but contains several original buildings in varied conditions. This is a marker on the site of the former colony commissary, located to the left of the administration building. Markers such as these are located throughout the DICE landscape today. The colony buildings and houses were designed by Little Rock architect Howard Eichenbaum. The farmhouses, designed in the colonial revival style, ranged from 3 to 5 bedroom floor plans and were one story with white frame and gabled roof and open porch. The administration building is a Greek revival style 2-story big construction with 1-story wings extending at the north and south elevations. This building features a 2-story portico supported by 4 columns with a Palladian style entrance that is flanked by the windows. DICE Town today encompasses 647 acres and contains a renovated administration building that functions as a city hall and museum. A memorial tablet for W.R. DICE, pictured here, was the geographic center of the town. DICE contains 30 of the original 500 homes in varied states of decay and alteration, and the Johnny Cash boyhood home, pictured here, is a 5-bedroom configuration home that is considered the best-known example of an authentically restored Federal Emergency Relief Administration colony home in the United States. The DICE landscape consists of Ditch 40 and the Tyronza River, pictured here, that forms the modern boundary of the town. The landscape today consists of farmland, gravel and paved road, and a mixture of occupied and unoccupied homes and buildings built in recent years in various states. DICE stands as a transforming landscape that is worthy of documentation due to its local, regional, and national significance for its role in the Great Depression and in modern agriculture. Thank you for listening to my presentation.